DNA sequencing, 3D bioprinting, and chromatographic techniques. GBU will be an attractive option to overseas researchers wanting to establish their research careers in India and will place Gujarat on biotechnology map of the world. Gujarat is emerging as a destination of choice, building robust ecosystems for innovation, research, technopreneurship, and excellence. Gujarat is geared to do all that it takes to be on the top. Come, join the journey of excellence. Come, experience Gujarat. Gujarat is home to a lot of biotechnology companies of likes of Zydus, Sun Pharma, Concord, Cadila, Intus, Hester, Zytex, JRF, etc., fueled by the talent pool from 50 universities. Gujarat also has government-supported as well as private research centers. The state government recognized the importance of biotechnology and established Gujarat State Biotechnology Mission (GSBTM) as the state node. GSBTM has been a research funding agency apart from supporting human resource development activities. In 2017-18, newer organizations have spun off, taking the development of biotechnology to a yet different level. Gujarat Biotechnology Research Centre (GBRC) was formed with state-of-the-art facilities to focus on translational research and technology development. GBRC has international collaborations with Royal Veterinary College and the University of Oxford. State has plans for developing BSL-4 laboratories, investing ₹250 crores to accelerate research. The state government has, with active partnership of central government, established Savli Technology and Business Incubator to support the technopreneurial startup ecosystem in Gujarat, for the startups to graduate into brownfield and greenfield spaces, and with a vision to develop a biocluster near Vadodara. The state government is also developing a biotechnology park over 223 acres of land with a CETP. GSBTM actively supports startups and companies through equity-based venture financing. To further encourage investments, the government of Gujarat has announced a very generous biotechnology policy 2022-27 that shall help the state in taking leadership. CAPEX support of 25% and OPEX support of 15% and other incentives are being offered under the Biotechnology Policy 2022-27. An expertly skilled workforce is the growth engine for knowledge economy. Hence, the state government is developing the world's first thematic university, Gujarat Biotechnology University, GBU, over an expansive 23 acres campus near Gift City, Gandhinagar, in deep collaboration with the University of Edinburgh, UK, especially in developing and curating bespoke curricula in key areas of biotechnology. Students will have an interface with latest instrumentation, including DNA sequencing, 3D bioprinting, and chromatographic techniques. GBU will be an attractive option to overseas researchers wanting to establish their research careers in India and will place Gujarat on biotechnology map of the world. Gujarat is emerging as a destination of choice, building robust ecosystems for innovation, research, technopreneurship, and excellence. Gujarat is geared to do all that it takes to be on the top. Come, join the journey of excellence. Come, experience Gujarat. Gujarat is home to a lot of biotechnology companies of likes of Zydus, Sun Pharma, Concord, Cadila, Intus, Hester, Zytex, JRF, etc., fueled by the talent pool from 50 universities. Gujarat also has government-supported as well as private research centers. The state government recognized the importance of biotechnology and established Gujarat State Biotechnology Mission (GSBTM) as the state nodal agency in 2004. GSBTM has been a research funding agency apart from supporting human resource development activities. In 2017-18, newer organizations have spun off, taking the development of biotechnology to a yet different level. Gujarat Biotechnology Research Centre (GBRC) was formed with state-of-art facilities to focus on translational research and technology development. GBRC has international collaborations with Royal Veterinary College and the University of Oxford. State has plans for developing BSL-4 laboratories, investing ₹250 crores to accelerate research. The state government has, with active partnership of central government, established Savli Technology and Business Incubator to support the technopreneurial startup ecosystem in Gujarat, for the startups to graduate into brownfield and greenfield spaces, and with a vision to develop a bio-cluster near Vadodara. The state government is also developing a biotechnology park over 223 acres of land with a CETP. GSBTM actively supports startups and companies through equity-based venture financing. To further encourage investments, the Government of Gujarat has announced a very generous Biotechnology Policy 2022-27 that shall help the state in taking leadership. CAPEX support of 25% and OPEX support of 15% and other incentives are being offered under the Biotechnology Policy 2022-27. An expertly skilled workforce is the growth engine for knowledge economy. Hence, the state government is developing the world's first thematic university, Gujarat Biotechnology University, GBU, over an expansive 23 acres campus near Gift City, Gandhinagar, in deep collaboration with the University of Edinburgh, UK, especially in developing and curating bespoke curricula in key areas of biotechnology. Students will have an interface with latest instrumentation, including DNA sequencing, 3D bioprinting, and chromatographic techniques. GBU will be an attractive option to overseas researchers wanting to establish their research careers in India and will place Gujarat on biotechnology map of the world. Gujarat is emerging as a destination of choice, building robust ecosystems for innovation, research, technopreneurship, and excellence. Gujarat is geared to do all that it takes to be on the top. Come, join the journey of excellence. Come, experience Gujarat. Gujarat is home to a lot of biotechnology companies of likes of Zydus, Sun Pharma, Concord, Cadila, Intus, Hester, Zytex, JRF, etc., fueled by the talent pool from 50 universities. Gujarat also has government-supported as well as private research centres. The state government recognized the importance of biotechnology and established Gujarat State Biotechnology Mission (GSBTM) as the state nodal agency in 2004. GSBTM has been a research funding agency apart from supporting human resource development activities. In 2017-18, newer organizations have spun off, taking the development of biotechnology to a yet different level. Gujarat Biotechnology Research Centre (GBRC) was formed with state-of-art facilities to focus on translational research and technology development. GBRC has international collaborations with Royal Veterinary College and the University of Oxford. State has plans for developing BSL-4 laboratories, investing ₹250 crores to accelerate research. The state government has, with active partnership of central government, established Savli Technology and Business Incubator to support the technopreneurial startup ecosystem in Gujarat, for the startups to graduate into brownfield and greenfield spaces. And with a vision to develop a bio-cluster near Vadodara, the state government is also developing a biotechnology park over 223 acres of land with a CETP. GSBTM actively supports startups and companies through equity-based venture financing. To further encourage investments, the government of Gujarat has announced a very generous biotechnology policy 2022-27 that shall help the state in taking leadership. CAPEX support of 25% and OPEX support of 15% and other incentives are being offered under the Biotechnology Policy 2022-27. An expertly skilled workforce is the growth engine for knowledge economy. Hence, the state government is developing the world's first thematic university, Gujarat Biotechnology University, GBU, over an expansive 23 acres campus near Gift City, Gandhinagar, in deep collaboration with the University of Edinburgh, UK, especially in developing and curating bespoke curricula in key areas of biotechnology. Students will have an interface with latest instrumentation, including DNA sequencing, 3D bioprinting, and chromatographic techniques. GBU will be an attractive option to overseas researchers wanting to establish their research careers in India and will place Gujarat on biotechnology map of the world. Gujarat is emerging as a destination of choice, building robust ecosystems for innovation, research, technopreneurship, and excellence. Gujarat is geared to do all that it takes to be on the top. Come, join the journey of excellence. Come, experience Gujarat. Gujarat is home to a lot of biotechnology companies of likes of Zydus, Sun Pharma, Concord, Cadila, Intus, Hester, Zytex, JRF, etc., fueled by the talent pool from 50 universities. Gujarat also has government-supported as well as private research centers. The state government recognized the importance of biotechnology and established Gujarat State Biotechnology Mission (GSBTM) as the state nodal agency in 2004. GSBTM has been a research funding agency apart from supporting human resource development activities. In 2017-18, newer organizations have spun off, taking the development of biotechnology to a yet different level. Gujarat Biotechnology Research Centre (GBRC) was formed with state-of-art facilities to focus on translational research and technology development. GBRC has international collaborations with Royal Veterinary College and the University of Oxford. State has plans for developing BSL-4 lab with a vision to develop a bio-cluster near Vadodara. The state government is also developing a biotechnology park over 223 acres of land with a CETP. GSBTM actively supports startups and companies through equity-based venture financing. To further encourage investments, the government of Gujarat has announced a very generous biotechnology policy 2022-27 that shall help the state in taking leadership. CAPEX support of 25% and OPEX support of 15% and other incentives are being offered under the Biotechnology Policy 2022-27. An expertly skilled workforce is the growth engine for knowledge economy. Hence, the state government is developing the world's first thematic university, Gujarat Biotechnology University, GBU, over an expansive 23 acres campus near Gift City, Gandhinagar, in deep collaboration with the University of Edinburgh, UK, especially in developing and curating bespoke curricula in key areas of biotechnology.
entrepreneurs, creating an integrated and supportive ecosystem for scientific and technological progress. Moving ahead right now, let me just give you a little bit brief about wildlife event. This event, organized by Gujarat State Biotechnology Mission, is centered on harnessing biotechnology for wildlife conservation and management. As the state's nodal agency for advancing biotechnology, GSBTM aims to integrate scientific innovation with on-ground conservation efforts to strengthen Gujarat's wildlife management framework. The program brings together senior official scientists and academicians and domain experts to explore biotechnology-driven tools and strategies that support wildlife health, disease surveillances, genetic diversity, and long-term conservation planning. The discussion highlights cutting-edge approaches across wildlife, livestock, human interfaces, One Health preparedness and diagnostic captive breeding and conservation genetics. So hence we all are here, as I said, initially mentioned, that we are here to definitely give our gratitude and honor to our delegates and dignitaries, more to it, to have a profound knowledge of wildlife conservation. Hence, it's time for me to welcome our dignitaries and felicitate them on the stage. So I'll initiate it by addressing Sri Ram Ratnanala, IFS, CF Wildlife Circle, Junagadh, and I request Dr. Mitali Dabi to kindly do the honor. Sir, I request you to please come forward.
I'll request dignitaries to come on the dais. To begin with, Pratap Singh Sahab, Dr. Ram Ratnanala. I'll also request senior faculty from Central University to also come on the dais. I'll also request Dr. Nishit Daraya, Director, Center for Wildlife, to come on the dais. Dr. Kanani, Joint Director, Animal Husbandry. Dr. S.K. Gupta from Wildlife Institute of India. Yeah. Thank you, one and all.
Now initiating the felicitation by Sri Snehal Bagatharia sir. He will be honoring Professor Pratap Singh Chauhan, Honorable Vice Chancellor, Bhakt Kavi Narsimha Mehta University, Junagadh. So can we hear a round of applause? Now, I request Dr. Mitali Dabi to kindly come forward and honor Dr. Ram Ratnanala, IFS, CF Wildlife Circle, Junagadh. Can we all together have a round of applause, please? Once again requesting Dr. Snehal Bagatharia to kindly come forward and honor Sri Amit Kanani, Joint Director, Animal Disease Control, Animal Husbandry Department. Now requesting Dr. Daksha Sakhya to kindly come forward and felicitate Dr. Nishit Daria, Director, Center of Excellence for Wildlife, BKNU. Now requesting Shri Hadmat Gadvi to kindly come forward and felicitate Shri Raghuveer Singh Jadeja, ex-forest officer. Now requesting Shri Rahul Kharadi to come forward and honor Dr. S.K. Gupta, Scientist, F. Wildlife Institute of India, Dehradun. Now I request Shri Devang Maru to kindly come forward and honor Dr. Apurva Singh Pawar, Scientist B, Gujarat Biotechnology Research Center, Gandhinagar. Now, once again, I would request our Dr. Snehal Bagatharia to kindly come forward and do the honors for Dr. Rajdeep Zala, Director, SBZ. Once again, requesting Snehal sir to please stay there as he has to felicitate Dr. Tomar, DLT. Please come forward. Ladies and gentlemen, let's not forget to put our hands together for all our dignitaries and delegates present over here across India, as they are here for us, as they are here to enrich this wonderful day for all of us by sharing their vision, mission, and objective, and their knowledge that they have acquired and sharing us and imparting the training to all of us. So once again, let me hear a huge round of applause for all our dignitaries and delegates sitting over here. And now, as the session begins, as the day begins, I would now request once again Dr. Snehal Bagatharia to kindly join us on the stage. He is our Joint Director, GSBTM, and he'll be doing the welcoming address for all of us.
Good morning, one and all. I heartily welcome dignitaries on the dais, off the dais, faculties, students, senior forest officials, officials from the veterinary department, officials from the information department, friends from press and media, I heartily welcome all of you on this very important event. I especially welcome Honorable Vice Chancellor, BKMU, Pratap C. Saab. He, consented and he gave us a time, his valuable time for this very important event. One of the eminent figure in wildlife research and one of the finest veterinarian, Dr. Ram Ratan Nala, CF Wildlife Circle, Junagadh. I also welcome Dr. S.K. Gupta, Scientist F, Wildlife Institute of India, Dehradun. I especially welcome Dr. Kanani, one of the eminent researcher helping in animal disease control across the state, whether it is veterinary or forest sector. Joint Director, Animal Husbandry, Dr. Kanani. I also welcome my colleague, Dr. Apurvasi Puwar, who has been working on One Health research and allied area. I heartily welcome Dr. Nishit, old friend of mine and colleague, Director, Center of Excellence Wildlife. And I also welcome other, faculties and friends, Dr. Suhas, Dr. Jani from Baudin Science College. All of you have been supporting this event very well. I also welcome Dr. Rajdeep Jala, Director SBZ, and Dr. Tomar for this very important event. I especially thank Jadeja Saab for providing all the guidance and support for developing biotechnology sector in the state. Under his able leadership and guidance, we have been doing various programs and we have taken this approach, to take the programs at a regional level, sector specific programs at a regional level, and series of programs are planned, and one of the event is wildlife event. And I also welcome all the students for tomorrow's event, which is on horticulture also. So thank you, sir, for all the guidance and support under your able leadership. GSBTM is doing, wonderful programs through your innovative approach. Once again, I welcome all of you. Thank you.
Thank you so much, sir, for your welcome address. And now we are looking forward towards Dr. Ram Ratnala, IFS, CF Wildlife Circle, Junagadh, to kindly share his words with all of us. Let's hear it for him, please.
Thank you, ma'am. Good morning to all present here. Dignitaries sitting on the dais. Chauhan Sahab, BC Bhakt Kavi Mehta University. Jadeja Sahab, one of my good friends, director of this mission. Dr. Gupta, scientist from WII, Amit Kanaji, Nisit ji, my colleague, Prashant, Rajdeep, R.D. Jadeja, Snail ji. So good morning to all present here. I would like to congratulate first Jadeja Sahab, director of the mission, to organize this beautiful event on wildlife management and research. So the mass who is sitting in front of us is the future of research and advances in wildlife management. I heard that students from different fields like veterinary, zoology, other aspects, other biological aspects, they all are present here, and definitely you all will be going to be benefited in upcoming today and tomorrow's lectures. There's a lot of people are here who are going to speak on different aspects. My dear friends, 2019, still I remember, all of us, we remember that Corona came. And Corona shown its power. 1945, when Second World War was there, and atom bombs were dropped on two cities, Nagasaki and Hiroshima, the number of people died were around 2 lakh each city. But the power of corona we all have seen. Millions of people died. So now the time has come up, friends, how we can be kept ourselves prepared, so that such corona incidences should not happen again, or if it happens, so how prepared we all are. Especially if I talk of the wildlife aspect, wildlife management. My dear friends, we all know that the population of wildlife is increasing day by day, year by year. The lion's population has increased from 674 in last census to now 891, almost 32% rise. Similarly, the population of leopards are also increasing. Similarly, the population of other wildlife, the smaller wildlife is also increasing. So all efforts because of the forest department and the people and the associated teams like the veterinary college and all, the efforts are clearly seen in the field. But at the same time, now what is happening is the interface with the public is also increasing, the interface with the animals is also increasing. You will see the movement of the lions when I was here in 2015 was in around 22,000 square kilometers area. 2020 is reached up to 32,000, and the recent census which was done, it was done in an area of 35,000 square kilometers, which covers more than thousands of villages of Saurashtra. So the kind of interface which is increasing, dear friends, that interface now, in past, all of us or many of us have read that many of the diseases which were hidden in wild animals, which were hidden in the dense jungle forest areas just because of the cutting of the forest for certain purposes, those animals, they came in interface with the humans. Interface बढ़ता गया ना, जंगल कम होते गए. So because of that interface, all those diseases which were hidden in wildlife or hidden in those organisms which were inside the deep forest areas and the interface was not that high, all those diseases, they came out. And havoc has happened. Many people have died. We have never heard the names of such diseases also, such viruses also, except what we have read in the books. They came outside the forest areas. Now similarly is happening with the other wildlife also. So they are moving out. Now it is the high time, my dear friends, that instead of going for the treatment part, what we were doing till now or few years back, we were focusing on the treatment part. Now we'll have to focus on the prevention part, how we can prevent it. Until unless we do not think about the prevention part, treatment is all okay. So now that high time has come that we should move on that epidemiological aspect where we can, read the prevalences, where we can read the incidences, if it happens, and the rate of prevalence and the rate of incidences. At the same time, how we can prevent these diseases these microbes to create diseases, exactly. So GSBTM definitely is, one of the biggest platform, and already many of the projects we have done with GBRC also, I think, which is the baby sister of GSBTM only. So biotechnology is definitely going to play a huge role, in managing the wildlife health and researches on the wildlife aspects. Through this particular platform, I would like to, to request Jadeja Sahab also we can have a kind of MOU or something like that, so that we can send our samples, regularly to the GSBTM. We can have a deep study on what is actually happening, with the microbes which are coming in the wildlife, or wildlife को कैसे impact कर रहा है, so that we can come up with certain strategies, we can come up with certain policies in which we can play an important role in preventing diseases inside the wildlife. So I would request from this platform, so that in the upcoming time, even with university also, we can play a major role, because I know such a huge mass is there, and you people are the future of the all these things. So let us work together. This wildlife wing, then the universities, then this mission, we all would have to come together, and we'll have to do something to prevent such incidences. Thank you so much. Thank you for giving me this opportunity of speaking at this place. Thank you.
Thank you so much, sir. It was indeed insightful. And now it's time to call upon the podium to Professor Pratap Singh Ji Chauhan, Honorable Vice Chancellor, Bhakt Kavi Narsimha Mehta University, Junagadh.
respected our mission director Digvijay C.G. Jadeja Sahab, equally respected our Ram Ratan Nalaji, our three speakers from today and tomorrow, Professor Director Nischid Daraya ji, Kanani ji, and Gupta ji, many officials from the state government and forest department, our university Professor Suhas Vyas ji, the professor and head, Baudin Science College Jani ji, many our others, faculty of professors, associate professor and assistant professor, our wildlife center, the internship, the students coming from the Surat, South Gujarat University, Saurashtra University, Baudin Science College, our Department of Bioscience and Wildlife. Dear, this is the program we connect with our center, the Wildlife Conservation Centers. The government has approved. It is only a one center given to a conventional state university. Mostly our task is very different. The university is working in a country, 1200 plus universities working in a country. And we have affiliated structure in the state university in the country. We have 50,000 affiliated colleges in the different university. And very close to 4.32 crores, means 43 million students, they are studying in the undergraduate PG program, MPhil and a PhD program. So it is a very complex higher education system in the country. Our higher education student is very close to two-thirds of population of UK, Germany and France. But then also, the question is always asking to our universities, why our university is not top 100 university of the globe. So the people are not asking to us, they are asking to, the top level. But what is happening in the grassroots level, that we have to understand. Because you see in the structure of the developed nation, mostly in the Europe and America, and we see the structure of this university, Princeton University, Harvard University, Cambridge. The span of the university is 150 years, 200 years, 300 years. And they sustain and maintain the standard of the higher education. We people, our gross enrollment ratio before 2014, very, very low compared to the third world, the nations. Now we are very close to 29%. Our gross enrollment ratio is concerned. And we reach to, at the time of 2047, the Viksit Bharat, the dreams of our Honorable Prime Minister, India will be a player for the third world largest economy, the China, America and India. That will be the structure. Today we are very close to 4.5 trillion dollar economy in the world. We are fourth and fifth. So far as the purchasing power parity is concerned, we are the third largest economy in the world. But so far the GDP is concerned, we are the fourth or fifth largest economy in the world. But at the same time, we have to understand what is the per capita income of the India. We are very close to $2,700 per person. That is the per capita income of the India. Now, where we have to do the task. India is a very young country. Our average age is very close to 29. So we have a competitive advantage in the next 25 years, where India will be lead to all over the world. Today, when news is, the published, and our government, the department is trying to reach our manufacturing sector contribution in the GDP in 2047 is 25%. At present, we are 17%. In our GDP structure, 65% GDP is coming from the service sector. It's not going to add the new jobs. And 17-18% from the agriculture sector and 17% from the manufacturing. We always compare to the Chinese economy. Chinese economy is $19 trillion economy. We are 4-5 times less. And population is very close to equal. So this is the, the task for our young and the university and our students. The average age is 29 and population, 65% population is below 35 age. It means we have a competitive advantage in this 25 year. And this era we have to task a very different dimension of the industry. Service sector, bioscience, biotechnology, where the India has a big potential, a market. And for that, the government has sanctioned the wildlife conservation, the Center for the Bhakt Kavi Narsi Mehta University. We are very close to the coastal area of this particular sort. The Devi Bhumi Dwarka, Porabandar, Junagadh and Gir Somnath, these 4 districts are covered by our Bhakt Kavi Narsi Mehta University. We have 246 colleges, affiliated colleges, and 110,000 students, particularly in the rural area of these 4 districts. So we have a potential students, and that's why our wildlife center, our Department of Life Science, and the botany, our Baudin Science College, we promote our wildlife conservation, where we are very close to Gir forest and up to the coastal area of the Gir Somnath. We are ready to work with the department. We are ready to work with the forest department, biotechnology department, and the respected our Digvijay Siji. We are very close to each other. We connect and do some MOU with our young students, because we need an internship. Where we go, the government has, passed the NEP 2020. The 4-year internship is given in the last year, the honors. You go in the field, study, work and publish the papers. Where we have to go, because we have a mass students. We are not on an average 1:15. Somewhere we are 300:1 teacher. So the structure of the Europe and America is different, and you have to understand the Indian, the structure is also different. We have a mass students, 43 million students. It means very big. You conclude and include the primary and secondary, then we are very close to 25 crore students in the country. It means very close to 20% of the nation population. So our challenges is very different, and that's why university is ready to work with the Department Biotechnology mission, and particularly we request to our Jadeja Sahab, you can do it and provide an opportunity to our students, our teacher, give some research project to the university, because university is ready to do this, because we have 2 departments, 1 wildlife center. We have appointed the director of the wildlife center, Professor Nishit Daraya is here. He is doing the research in the field. So we connect the particular people, the working in the field, and we are studying in the theory in the classroom. We apply theory to the field, and field, whatever the experience is there, that we can connect to our classroom. That is the need of the higher education. That's why we are not in the top 100 university. Our IIMs, our IITs, these are also, coming to the 168, 170, 172, where we consider the topmost institute in the country. Our problem is we are not connect the people from the foreign. Our student ratio is 90% student from our own. because our demand, our consumption pattern is whatever we produce, 90% we consume. So whatever the term tariff is there, but it is not highly affected in the last 6 months in the country because we consume our own product. That's why the people, because the $30 trillion economy is there, that's why it affect. After the 6 months is over, we will face some issues, but we are the fighter. India is a fighter. Indian young mind is a fighter. And our generation Gen is very sober and very intellectual. It's not like our neighbors. We people attack, and we attack in the intellectual property. We attack in our knowledge. We reach to a pull, and that pull will be a work for the Viksit Bharat at 2047. So my blessings, our 2-day interaction, today and tomorrow, the people at the keynote address, they can talk in the field, and whatever the outcome is there, we provide to our director, the mission director, Digvijay, and then, if it is possible, university, now this is the span. Our university is a very new university, just a 10-year pass, and building and infrastructure is going on. Within 1 year, everything will be available to our university. The full facility, whatever the university of 60-year-old, the same university, this university also work in the research, teaching, and extension, and we are ready to provide a facility, expertise facility to the field. So thank you very much, and the best wishes to my student. They are coming from the department to department, wildlife student and Baudin Science College. We will be the work together. In case you have any query, you need any facility, university is ready to provide all facility to my young student. Thank you very much. Bharat Mata ki Jai, Vande Mataram.
Thank you so much, sir. Your words not just hit our minds, but also ignited the spark for the vision that we are looking forward. Thank you so much. And now it's time to call upon the stage or on the podium our mission director, Shri Digvijay Singh Jadeja, IAS, a man with a vision, walks ahead not with certainty, but with courage, strong enough to light the way. He doesn't just see the world as it is, he sees what it can become. His clarity is his compass and his conviction is his strength. So let us all put our hands together for our sir. Can we hear some huge round of applause, please? Thank you so much.
સૌને નમસ્કાર. ખૂબ જ અનુભવી એવા આપણા વીસી, આ ત્રીજી યુનિવર્સિટી છે, સરકારશ્રીએ એમની પ્રતિભાને પારખીને, એટલે આપ નસીબદાર છો કે આટલા અનુભવી વાઇસ ચાન્સલર પ્રતાપસિંહ ચૌહાણ સાહેબ આપને મળ્યા છે. યુનિવર્સિટી ભલે 10 વર્ષ જૂની હોય, પણ બાઉદીન કોલેજના લગભગ વિદ્યાર્થીઓ કેટલા છે અહીંયા? 1905માં બનેલી કોલેજ છે. 120 વર્ષ જૂની કોલેજ છે. અને લગભગ જૂના જે કોઈ આપણે મળીએ ને તો એક એને ગૌરવ હોય. અને વેન આઈ વોઝ એસડીએમ જુનાગઢ, ત્યારે એના 100 વર્ષ પૂરા થયા હતા. ઉજવણી વખતે પણ હું ત્યાં એઝ એ ગેસ્ટ એ કોલેજમાં હું આવેલો હતો. એટલે આપણી પાસે ખૂબ અદ્ભુત વારસો છે, પણ એને કેવી રીતે આગળ લઈ જાવો એના માનવ મંથન માટે આપણે ભેગા થયા છીએ. માનનીય પ્રતાપસિંહ ચૌહાણ સાહેબ, ખૂબ જ ઉમદા અધિકારી એવા ડૉક્ટર રામનતન નાલા સાહેબનું અહીં પોસ્ટિંગ થયું ત્યારે બધા યાદ હતા કે બહુ સારા ડીસીએફ હતા, એટલે સાહેબને હજી લોકો આવીને એ જ યાદ કરે એમને જે કામ કર્યું એ. શ્રી અમિત કાનાણીજી, ડૉક્ટર નિશિત ધારેયાજી, અમારા ખૂબ વડીલ એવા રઘુવીરસિંહ જાડેજા. ગયા વર્ષે આપણે હતું કે બહુ ફ્લાવરિંગ સારું થયું હતું આંબાનું, અને પછી એકદમ એમાં રોગ આવી ગયો હતો, તો બધે બહુ નુકસાન થયું હતું. તો જાડેજા સાહેબનું પોતાનું ફાર્મ છે, પોતે સાયન્ટિફિક રીતે આંબાને બીજા ફળ પણ રાખે. તો એમને કોઈ નુકસાન, ત્યારે મેં એમને પૂછ્યું હતું, તો એમને સાયન્ટિફિક એવા સમજાયું કે આવું કરવાથી આ નુકસાન ના થાય. તો આજે એમનો પોતાનો લાભ છે આખા વિસ્તારને મળે, એટલા માટે આપણે ખાસ એમને આમંત્રણ આપીને આજે અને કાલે પણ એમને આપણે બોલાવ્યા છે. એવા મારા વડીલ મિત્ર રઘુવીરસિંહ જાડેજા સાહેબ, શ્રી S.K. ગુપ્તાજી, ડૉક્ટર અપૂર્વસિંહ અને માય ઓલ્ડ ટીમ તોમર સાહેબ એન્ડ ઝાલા સાહેબ, આપ સર્વેનું અને જે આટલી વિશાળ સંખ્યામાં બધા વિદ્યાર્થીઓ, ચૌહાણ સાહેબનો ખાસ આભાર કે બધાને આજે ચાલુ કોલેજનો એકેડમી ચાલુ છે છતાં પણ બધાને અહીંયા હાજર રહેવાની છૂટ આપી. એવા મારા વિદ્યાર્થીનીઓ અને પત્રકાર મિત્રો, અમારો ફોરેસ્ટનો બધો સ્ટાફ અને મારો સ્ટાફ. ગુજરાત સ્ટેટ બાયોટેકનોલોજી મિશન એટલે કામ ઘણું કર્યું છે, પણ જે એક સેન્ટ્રિક આપણે કહીએ ને એવું, એમાં કોઈ ટીકા નથી, પણ એમ અમુક જે યુનિવર્સિટીઝ જે છે, જૂની યુનિવર્સિટીઝ છે જે મધ્ય ગુજરાતની હોય, એના સ્ટુડન્ટ્સને હોય, એને એનો ખ્યાલ હોય ને રિસર્ચમાં એ ભાગ લેતા હોય. આખા ગુજરાતનું આપણે જોઈએ તો આપણો આ કોસ્ટલ બેલ્ટ છે, કે રાજકોટ ઇવન છે, કે નોર્થ ગુજરાત છે, ટ્રાઇબલ બેલ્ટ છે, એના આપણા વિદ્યાર્થીઓ જે છે એ એટલે વિદ્યાર્થીઓ આવી જાય છે, એટલે એવું ના વિચાર કે સાહેબ ખાલી વિદ્યાર્થીઓ જ કહે છે. એના વિદ્યાર્થીઓ છે એટલું જે રિસર્ચમાં એમને ભાગ લેવો જોઈએ, જે એમને તત્પરતા હોવી જોઈએ. કદાચ અમારી એમાં કોઈ ક્ષતિ હશે કે અમે તમારા સુધી પહોંચી શકતા નહોતા, એ જોઈએ એટલો ફ્લો એમાં જોવા મળતો નથી. તો આજે અમે આ ત્રીજો ડિસ્ટ્રિક્ટ છે, જિલ્લામાં જવાનું કારણ છે કે આપણા વિદ્યાર્થીઓ છે એની સાથે સીધું અમે કનેક્ટ થઈએ અને એમને પ્રોત્સાહિત કરીએ કે તમે કોઈ સારું રિસર્ચ કરો. સરકારશ્રીની ખૂબ સારી સ્કીમ છે. આપણા દિગ્દર્શક વડાપ્રધાનશ્રીએ 2004માં મિશનની સ્થાપના કરી, એટલે 21 વર્ષ પહેલાં. અને આપ તો આ વિષય ભણો છો ને આપ તો બધા વિષય નિષ્ણાંત છો, એટલે આનું ભવિષ્ય ખૂબ ઉજ્જવળ છે, આપ જાણો છો, મારે એમાં કાંઈ બોલવાની જરૂર નથી. પણ આનો મેક્સિમમ, હવે ધારો કે ગીર ફોરેસ્ટ છે, એના પણ હશે, સાહેબે જેમ કીધું ને, અને અમે તૈયાર છીએ, વી આર રેડી, અમે MOU કરવા માટે એટલે જ અમે આવ્યા છીએ કે આપ સારા કોઈ પણ પ્રોજેક્ટ અમને આપશો. અમારું કામ જ ફંડિંગનું છે કે સારો કોઈ પ્રોજેક્ટ આવે તો એને પ્રોપર ફંડિંગ કરવું ને એને સારું વાતાવરણ પૂરું પાડવું. અમારા ડૉક્ટર સ્નેહલ છે, ઇઝ ઇન્ચાર્જ ડાયરેક્ટર ઓફ GBRC, તો એ પણ તમને ખૂબ એમાં મદદરૂપ થશે. ને કોઈ પણ એમાં હોય કે એ આપણે પ્રોજેક્ટ હશે તો એને આપણે 100% એને આપણે આગળ. આ તો નેટવર્ક પ્રોજેક્ટ કે ભાઈ ડિપાર્ટમેન્ટ કોઈ સારો પ્રોજેક્ટ મૂકે અને એ કરવાની વાત છે કે ભાઈ નાલા સાહેબે કીધું કે એ કોઈ સારો પ્રોજેક્ટ આપણને આપે તો અમે 100% કરશું. યુનિવર્સિટી પોતે કોઈ સારો પ્રોજેક્ટ લે, હોર્ટિકલ્ચરનો હોય કે કોઈ વાઇલ્ડ લાઈફનો હોય. અને સેકન્ડ થિંગ સાહેબ, અમે આવતા અઠવાડિયે 20 તારીખે પોરબંદરમાં અમે આપણો જે 2300 કિલોમીટર લાંબો દરિયાકાંઠો છે તો એનો જે ઉપયોગ થવો જોઈએ એવો થયો નથી. તો એમાં બાયોટેકનોલોજી જે ઘણા બધા એમાં વ્યાવસાયિક ધોરણે પણ થઈ શકે એવું છે. આપણે કહીએ કે ભાઈ આપણો દરિયો પ્રદૂષિત થાય છે, ટેમ્પરેચર વધે છે, માછલીઓ છે એ મરી જાય છે, મળતી નથી, માછીમારી સંકટમાં છે, બધા જાણીએ છીએ. તો આપણે શું કરવાનું? તો એના માટેનો એક સેમિનાર મેં 20 તારીખે પોરબંદરમાં રાખ્યો છે, કોસ્ટલ આખા રિજિયનનો. એમાં પણ જો સાહેબ આપ અનુકૂળતા કરો અથવા તો આપ કોઈ આપણા ફેકલ્ટી ડાયરેક્ટર ઓર સ્ટુડન્ટ્સ કોઈને ઇન્ટરેસ્ટ હોય એમાં, જેને મરીન બાયોલોજી કે બાયોટેકનોલોજી, તો એ પણ દે આર મોસ્ટ વેલકમ. અને એ લોકો આવે, એમાં ભાગ લે. ને એવું નથી ખાલી સાંભળવા જવો, તમારી પાસે કોઈ આઈડિયા હોય તો અમને કહો. આજે આ વિષય મારે તો નવો છે, અમે તો વહીવટી અધિકારી છીએ એટલે અમને તો જ્યાં જાય અમે તો અમે સેટ થઈ જાય એવું હોય. એટલે હવે તો પછી વિષય નિષ્ણાંત ન હોય તો એ કંઈ પણ અમારું પ્રદાન કરી શકીએ, અમારામાં ઇનબિલ્ટ હોય છે. કેમ એમ કે ભાઈ અમે ક્યાંય પણ જાય અમને જે વિષય બોલે અમે બોલવા માંડીએ એમ. એટલે આ લોકોને ખબર છે મને નોટ નહીં આપે, મેં કીધું એવું મને ટેવ નથી. હું 7 મહિનામાં જેટલું સમજ્યો છું એ હું તમને કહીશ ને હું તમારી જેમ આ લોકો જે બધા નિષ્ણાતો છે એની પાસેથી હું પણ કંઈક મારું જ્ઞાન વધે એવું કરીશ. પણ આપણો ખાલી સેમિનાર 2 દિવસ આપણે વાતો કરીએ અને છૂટા પડી જઈએ સારું બોલીએ એના માટે નથી. આપણા અહીંના જે વિદ્યાર્થીઓ છે કોઈ સારું રિસર્ચ પેપરમાં કોઈ સારું રિસર્ચ કરે, અમારી પાસે આવે, એપ્લાય થાય, અમે 100% એમાં એમનો એ પ્રોજેક્ટ મંજૂર કરશું. ખૂબ સારી સ્કીમ છે, 3 વર્ષ માટે આપણે એમને ફંડિંગ પૂરું પાડીએ છીએ. કોઈ એમાં કેપ નથી કે ભાઈ આટલું જ ફંડ મળશે. તમારો પ્રોજેક્ટ સારો હશે તો એની ટેકનિકલ એડવાઇઝર કમિટી છે એમાં હાઈ લેવલના સાયન્ટિસ્ટ છે એ એપ્રૂવ કરશે. એમાં કોઈ ત્રુટિ હશે તો એવું સીધું રિજેક્ટ નહીં કરે, તમને ગાઈડ કરશે કે ભાઈ તમે આટલું આટલુંમાં સુધારો કરીને લાવો તો તમને અમે મંજૂર કરશું. અને 3 વર્ષ માટે તમામ સુવિધા, હવે તમારું યુનિવર્સિટીનું આવો સરસ વીસી છે, આટલા સારા ડાયરેક્ટર ને બધા છે, તો મેઇન છે એ યુનિવર્સિટી કેવું વાતાવરણ પૂરું કરે. પ્રોજેક્ટ મંજૂર થયા પછી એની લેબ ફેસિલિટી યુઝ કરવા માટે કે XYZ જે કાંઈ પણ બીજી ફેસિલિટી યુઝ કરવા માટે કેવી તત્પરતા દાખે, એ તો તમારે પ્રશ્ન નથી, એ તો તમને પૂરું સારું તમને વાતાવરણ મળવાનું જ છે. તો આજે એવા અહીંથી તમે લોકો UGPGના જે વિદ્યાર્થીઓ હોય અને કોઈ એમાં આગળ આવે તો અમને ખૂબ આનંદ થશે. વાઇલ્ડ લાઈફમાં પણ સાહેબે કીધું છે MOU કે કરવું હોય, જે સાહેબ આપ પ્રપોઝલ લાવશો કોઈ સારું આપણે કરવા માટે અમે તૈયાર છીએ. આજે આવતીકાલે હોર્ટિકલ્ચરનું છે, આપણે કાયમ કરીએ છીએ કે ભાઈ આંબામાં આવે છે કે ઓલી વ્હાઇટ ફ્લાય આવે છે, આ થાય, તો એને અટકાવવા શું કરવું જોઈએ તમારા હાથમાં છે. અને કેમ કોરોના એમ નવા આવશે જ, પ્રાણીઓમાં પણ આવશે, બાગાયતમાં પણ આવશે, માનવી પણ આવશે. તો એ ભવિષ્યના રોગો શું છે ને એના કેવા પ્રકારનું રિસર્ચ કરી શકાય ને એને અટકાવી શકાય, એ માટે એ દિશામાં આપણે આગળ વધવાનું છે. આપણે મેં પહેલાં કીધું એમ કે આપણો ગોલ હોય ને, સાહેબે કીધું ને, સાહેબ એઝ એ VC એમને હોય કે ભાઈ ટોપ 100માં યુનિવર્સિટી આવે. એમને તો એ તમારા વડા છે, ઇઝ લીડર, તો એને તો એ જ હોય ને કે ભાઈ લીડર હંમેશા એવું છે કે ભાઈ મારું એમાં આગળ વધાય. તો એ પોતે એકલા જ જવાનું, એ તમે બધા એમાં સહયોગ આપશો તો આ આખું ઊંચું આવશે. બે બાજુનું છે, એમાં જ્યાં કાંઈ વહીવટી કક્ષાએ ખૂટતું હશે તો એમની જવાબદારી છે, પણ જ્યાં સુધી આપણા વિદ્યાર્થીઓમાં એ પ્રકારનો આત્મવિશ્વાસ નહીં આવે કે ભાઈ અમે પણ કાંઈ કરી શકીએ છીએ, અને આપણે ટાર્ગેટ આપણો શું હોવો જોઈએ કે સારું અભ્યાસ, સારું રિસર્ચ કરીએ અને સારું બનીએ. વાં પહોંચવું છે, અહીંયા જવું છે, ફોરેન જવું છે, એ તો તમારામાંથી તો આપોઆપ કોઈક લઈ જશે, તમારે મહેનતે નહીં કરવી પડે. પણ એના માટે જે સક્ષમ બનવાનું છે એમાં જો ધ્યાન આપશો ને તો તમને લેવાળા તો હજારો મળી જશે. બાકી આપણે ટાર્ગેટ રાખીએ કે મારે સીધું જઈને વયું જવું છે તો તમારે લાઇનમાં ઊભું રહેવું પડશે. ઓલું તમને લેવા માટે લાઈન લાગવી જોઈએ કે ભાઈ ચાલો આ ભક્ત કવિ નરસિંહ મહેતા યુનિવર્સિટીના છોકરાઓમાં આટલું પોટેન્શિયલ છે, આટલા સારા રિસર્ચ પેપર તૈયાર કર્યા છે, તો આપણે ત્યાં જાય, કેમ્પસ ઇન્ટરવ્યૂ થાય ને લઈ જાય કોઈ તમને. આ બધા અધિકારીઓ છે લગભગ તમારો જેવું જ બેકગ્રાઉન્ડ હશે, આ ફોરેસ્ટમાં IFS બધા છે સાહેબ ને બધા અમારા મિત્રો, તેનું બેકગ્રાઉન્ડ તમારો જ અભ્યાસ એ જ હશે, એમાંથી જ આગળ આવ્યા છે. અમારા બધા ડૉક્ટરો છે, અમારા બધા જેટલા અમારો સ્ટાફ છે બધા ડૉક્ટર છે, એટલે અમે જાય તો મને ઓળખાણ એમ જ આપે, એમ આ ડૉક્ટર હશે એવું. પણ ખૂબ સારું પોટેન્શિયલ છે અમારો આજનો આવવાનો એ જ છે. હું કોઈ તમને વિષય ઉપર તો મારું એવું નથી કે તમને હું જ્ઞાન આપી શકું, પણ તમે એવું સારું, તમને મારી અપીલ છે કે તમે એટલા સક્ષમ બનો કે તમને લોકો સામેથી લઈ જવા માટે આવે કે તમે અમારી ઇન્ડસ્ટ્રીમાં આવો, કે ફાર્માવાળા આવે, કે ઇવન ફોરેન કન્ટ્રીવાળા તમને સામેથી લેવા આવે કે છે. કોરોના આવ્યો તો એમ્સના છોકરાઓને ચાર્ટર્ડ પ્લેનમાં લઈ ગયા હતા, આપણા જે છોકરાઓ હતા એને ત્યાં USA વાળા, એને કેટલાય મારા જાણમાં છે કે ભાઈ આ છોકરાઓ છે એનામાં પોટેન્શિયલ છે, એની પાસે આપણું છે કે એમ્સનું મોટું છોકરો છે અહીંયા કામ આવશે, એ લોકો સામેથી લઈ ગયા હતા. તો એવું કોઈ સારું આમાંથી તૈયાર થાય અને સાહેબના કાર્યકાળમાં જ થાય, અને બંને સાહેબોને જે એમને કીધું છે એ પ્રમાણે કાંઈ પણ કોઈ પણ જાતનો અમે તો સામેથી ઉઘરાણી કરશું મારા સ્વભાવ પ્રમાણે કે સાહેબ તમે કંઈક લાવો અને આપણે એમ આગળ વધશું. આપ બધા સમય કાઢીને આવ્યા છો, તમામનો ખૂબ ખૂબ આભાર, ધન્યવાદ.
Thank you once again, sir. And it's the moment where we'll be starting up with our technical session. So I request all our dignitaries to kindly take down your seats on the first row at the sofa. And also I request the hospitality team to kindly clear the stage as soon as possible. And now initiating this wonderful technical session, which will be conducted by Shri Amit Kanani, Joint Director, Animal Disease Control, Animal Husbandry Department. So the topic gonna be on biotech approach to the livestock's human interface, innovations in disease surveillance for One Health preparedness. And to all of you, those who are present over here, as we mentioned that this will be a very knowledgeable session. So all the points that indeed need to be asked, I request everybody to jot it down, to pen it down whenever the sessions are be taken by our dignitaries, by our esteemed delegates. So it is your responsibility to jot it down and pen down all the important notes, all the important queries which can be taken care of later. As we start with the technical session, this is going to be the most interesting segment of this beautiful day. And once again, thanking all our dignitaries, those who have marked their presence at this beautiful day. So the moment dignitaries arrive and they take down the seats, we'll begin with the technical session. So two dear students, those who are present here, this is for them. The session gonna start. But once again, notifying everybody, firstly, that this session is indeed for all of us. The sessions which are coming up now, they are not just profound to listen or acquire the knowledge, but also to inherit them in our lives. So hence, let's all together call upon Shri Amit Kanani ji, the Joint Director, Animal Disease Control, Animal Husbandry Department, to start up with the first session of this day. And let us all together put our hands to welcome Amit sir as he'll be taking the first session of this day.
Respected Jadeja sir, in his leaderships, those seminars and symposia happening in the Gandhinagar and Ahmedabad, its brick comes to the real at the field level, and really we proud, not our only Gujarati, but throughout the India, the areas of the Lion, Sasan, Gir. And I'm thankful to the GSBTM for inviting me and deliberating my experience during my careers in animal disease control and wildlife disease investigations, whatever the brings these things. And it is really now very much needed to use the biotechnology weapons. We know as the disease scenarios are day-to-day changes. And yes, Dr. Ramratan Nala sir, IFSS rightly says, during the COVIDs, we lost so many human beings. Not only during the COVIDs we lost human beings, but disease was also transmitted to the many other species also. They are jumped to the other species. We know COVID, but before that COVID we have prepared for the avian influenza or a high pathogenic influenza. We already started work on surveillance on the avian influenza because Gujarat is not only land of the lions or gir forest, but there are so many migratory birds, particularly now winter season is started. They are visiting the Gujarat. Along with brings many viruses apart from different countries. They are traveling and they are nestings in our wetland areas like Nal Sarovar is there and many other areas. So there is a chance of disease spreading. As we know, COVID within 2 months of this year, COVID is presence in all the places. But this virus is how they jump. Before this COVID, there is a SARS-CoV virus is also there. Middle East respiratory symptom viruses. This is also a coronavirus. This coronavirus is already present in the Middle East. So far, it is also reported only in camel and from camel to the human beings. So many lives we lost, human lives, due to the MERS-CoV. That is Middle East respiratory syndromes, which is happening in the camel, and from camels to it transmit the human being. So we know the disease transmission panels, and there is a pandemic. As sir says, Gujarat Biotechnology missions is full open to the students and new innovators to bring their ideas and to do the research on certain areas. On the other hand, international agencies like WHO, World Bank has supported 2 billion rupees to the India for pandemic preparedness funds. So fund is now not a limitation, either is from the government of India, World Bank agencies, food and agriculture agencies, but now we have to innovate the ideas. What we see in the fields, we bring into the research. So with this beginning, why? As we know, wildlife populations slowly due to the very good conservation status, it's increasing. People now more take care about the wildlife. Instead of hunting, they are now loving the wildlife. On the other hand, human populations are also bringing. So due to these interactions, there are many new diseases are emerging, like Nipah viruses is there, avian influenza viruses is there. Recently in last week, Crimean Congo hemorrhagic fever is reported in the children's, where in the Savarkundla villages. This is highly fatal diseases. Of 135 cases so far reported since 2011, most of cases are reported in the Gujarat. Why? Virus know the political boundaries? No, virus not the political boundaries, but we Gujarat have established a strengthened surveillance systems. Any hemorrhagic fever diseases reported in the humans, it is immediately confirmed with the new biotechnological tools like PCR and real-time PCR, and we also monitoring those things. So this way, Gujarat is leading in control many diseases. Similarly, there is a Bio-Resilience conservation is really needs for the public health. Why? There is a risk factors are increasing. Because increasing populations, we are now constructing buildings and concretes and interfering in the jungles and other areas. In the same change, there is a climate change due to the pollutions and other things. We extreme weathers and different type of the rainy seasons. We recently saw the unwanted monsoons in the particularly central, and lots of grazing areas, grasses are lost due to these things. Even there is a global trades and global traveling. Diseases are move very fastly. On the other hand, people having a increasing money, wealth and everything, they having increased the craze of taking some unwanted species with them as a pets. Previously, we just kept as a cat and dog as pet. Now many people use many wild type animals in the premise. So there is an increasing bonding between species. Those we are only a wildlife species, now they brings to the home. As the climate changes, vector populations like mosquitoes and ticks and mites, they are now become changing. And due to this, and due to the compensate this human population's hunger, there is an intensified livestock farming is going on. Previously, there are only few people kept poultry at their home. Now there is a bigger poultry farmings, particularly in the Anand area is there, Palanpur is there. The big poultry farmings are people. People keeps intensive poultry farmings. So to cope up the food requirement, protein requirement, large dairy farms are now coming with crossbreeding and other. We are importing many animals from the countries to increase our breeding centers. So due to this, and now also people keeps horses for the good venture. So due this climate change, vector population change, diseases are now jumping and new diseases have started, like Japanese encephalitis is there. Or once these viruses are stable in the environment, if we are not rightly diagnosis those viruses, their genetic makeups, they may start stabilize in the environment and there may chance to go to the human, and once it's go to the human and there is a chances to human transmissions, and we can definitely get another pandemic like COVID-19. So there is a unified vision. Jadeja sir has rightly said, and they are even in leaderships, the right goals of GSBTM. They are not focusing in the one areas, they are focusing in the horticultures and all wildlife sectors, genomic sectors, all the areas. Because we have to now do the unified approaches to control those diseases, to maintain those diseases. So when there is a wildlife, livestock and human health is concerned, they are the interrelated. If any diseases are there, like we discuss in later on, foot and mouth disease and other things, they will definitely affect the prey animals like deer and sambar, and this will definitely affect the lion health. So One Health framework, what we say One Health? One Health means not to work in the silos. All the different line departments, all the different expertise, working in the different fields come together. And here, the expertise from the biotechnology came with their innovations, came with their new ideas, and they give solutions to different departments, different line departments, either it's a horticulture, it's a agriculture, genomic crops, cottons. So this is the way we can integrate all the surveillances. And previously the surveillance side is different. In the animal husbandry, we do the sero monitoring and sero surveillancing using antibody titers. But now with the genomic tools, we using PCR, real-time PCR, digital PCR facilities, we surveillance the different viruses among the different things. So these biotechnology approaches bring together to do the more responses. So molecular technologies now become a forefront weapons to identify any diseases, any pathogens, their molecular epidemiology, genomic trackings. I will give the one example. In 2008, there is a suddenly start of equine influenza viruses infections in the Gujarat. At the same time, there is in Australia, equine influenza viruses cases are reported in the Australia. So we think that there might be a chance of this virus came from the Australia to the India. But when we do the whole genome sequencing and studies, we already have a vaccinations with us in 2008 for equine influenza. But when we do the genetic analysis, virus is not matched with the Australian strain. Current available vaccines are not matching with the new viruses. And when phylogenetic analysis of those equine influenza viruses is done, that was matching with the Chinese and Mongolian viruses. And when we started the study the line cases one-to-one, and that it was noticed that this disease was started from the Katra, Jammu, and it slowly spread to the and come to the Gujarat. So this genomic analysis not only give the ideas, but it also give how the viruses are circulating among the various populations. Simple tools. Right now, we bring for the educations and other purposes, penguins are kept at the Science City. This is also under the leaderships of the Department of Science and Technology. Merely you see the penguins, even I am a veterinarian working since long, but I cannot identify it, it's a male or female. Okay, similarly in many birds, we could not, even in emu, we could not identify it. Simple PCR technology, a biotechnology tool immediately says, even from the feather of birds, we can say it's a male or female. This is the primary technology to lots advanced microarray systems. When we are studying during the colleges, there is a progeny testing programs. We all know this is the area of Gir breeds, our Gir cattle. When we go for the breeding of the Gir cattle, we have to keep records for selections of the good females or good male, because male is the dominant, and from one male we are doing the artificial insemination and many doses. So selection of male is very important. At that time we keep the records of parents, sires and everything. Now biotechnology make is a very strong. On the day of the birth of the cattle, we know what will be the proposed milk production of that cattle. And this is also possible with the support of GSBTM. They do the genomic breeding value, GBV, with using the microarray chips with NDDB and other forms. Similarly, we see there are lots of stray, especially male animals. Male animals are not more useful. And many cities, and especially in municipal corporations, males are stranded on the roads and there are accidental and there are many cases. So in this cases. Now sex cement technology are coming, cements are sorted, and these sex cements, once we say, yes, this is the only Y chromosome or deleted and only X chromosome cement, how we can measure the quality of those cements? These biotechnology interventions give, now we have our Make in India sex cement technology. Previously, each cement dosage was around 800 rupees to 900 rupees. With Make in India, we bring it to the 300. And with the government give subsidy to only 50 rupees. So these are the biotechnology interventions we make our breeding and easily. On the other hand, when we say the pandemic preparedness, many influenza viruses, foot and mouth disease viruses, before 2 years we hit break with the lumpy skin disease virus. So GBRC, with the help of this biotechnology surveillance tool, they survey where in which areas new viruses is coming. Because wildlife sectors brings new viruses, so during their fecal samples, feather samples they are studying, along with what are these birds are new viruses coming. So this type of surveillances, non-invasive surveillances, where you do not need to catch the birds, just from their fecal droppings and other things, they surveillances and proposed the exposure, and that will help us in preparedness and control any outbreaks. These are the different sites where the GBRC is collecting samples. Similarly, foot and mouth diseases. Previously in foot and mouth diseases, when there is an outbreak is reported, we are collecting samples. Now, new tools are developed. Right from the dairy cooperatives, we know the Gujarat is a very much powerful in the cooperative sectors. From the dairy cooperatives, you go the Mahi dairy, from different cans, even from the tankers, you can identify there is a foot and mouth disease is there or not, any villages. So how many villages are affected? Right from the milk, you can address the disease prevalence rate in the districts. And even before the clinical sign appears, because disease identifications is very important, early disease identifications is most important to contain any diseases. Once diseases spread like lumpy skin diseases, we are little bit late and it spread throughout the country. So early detections, before 4 days clinical sign appears in the animal, foot and mouth disease can identify from the milk with new technology. And even with the copy numbers with the digital PCR facilities that are available at the GBRC. So these are the non-invasive samples, we do the lots of works. For bioconvergences, monitoring different diseases, it is very important. Preventing diseases, transmission from one sector to another sectors, from livestock to the wildlife, it is very important. There are genetic tools for preservations of the DNA, barcoding DNA, because DNA is the most resource material, and biodiversity we can identify with the DNA markings. Here with the genomic value, when I will say regarding the GBV or breeding purifications, even a blood sample of the cattle, even identified, you go to the lab, that will be say this is the blood from Kakhraj cattle having this type of the skin colors, having this type of the milk production traits. This is the samples from the, even people, scientists sitting in the laboratory can say which breed of these cattle are there. So this type of the fingerprintings are very important now, and for health, the biotechnology is now necessary. Definitely, when I say in the very first lectures, many diseases now change due to the ecology and diversification changes. They are jumping to the species. Canine distemper is a primary disease of the dog and canine family. That was few years back, jumped to the feline families. And our big cats are at the affected. Similarly, when we say the avian influenza, so far avian influenza is only restricted to the birds and few human cases are reported. But recently, due to the change in their molecular structures, avian influenza is also reported in the cat and also reported in the cattle in the Mexico and USA. And owners or workers working in the cattle farms, they are also get infections. So it is very important. On the other hand, rabies is another thing. We in India, so far we have not reported bat-mediated rabies viruses. But there are a lot of rabies viruses are there. Rabies virus is regularly maintained in the wildlife circulations, and when these wildlife circulations, these wildlife animals come in close contact with the urban or peri-urban areas, this rabies virus jump to our domestic dogs and cats, and from domestic cats and dogs, many human bites things are happening, and India is on the top countries where most dog bite cases are reported and many deaths are reported due to the rabies. But yes, biotech it is not merely easily to catch each and every wildlife populations and dog populations and get them vaccinated with the injectable vaccinations. But yes, there is a way forward. There is a way in the futures. We can do collaborations with the innovations in the different vaccines. Oral vaccines can be prepared for the rabies and other infections. So we can deliver those oral dog. But there is a futures for canine distemper virus vaccines for the felines and other animals, oral vaccinations, AI-driven biosurveillances, okay, increasing awareness among the peoples. Their participatory approaches are also required, that is called PPE models are there, and strengthening resilience in the wildlife, livestock and human interfaces. All three populations are day by day increases. Thank you very much. Okay, any questions or we have to take last? મિત્રો સ્ટુડન્ટો કઈ ક્વેશ્ચન્સ હોય તો વેલકમ. કોલેજનો જ ક્લાસ ગણજો, બરાબર છે. જાડેજા સાહેબ સ્પેશિયલ આપણા માટે અહીંયા લઈ આવ્યા છે, એટલે ખાસ કરીને કોઈ પણ ક્વેશ્ચન ઊભો થાય તો પૂછી શકો છો. અધરવાઇઝ જ્યારે પણ આપણી બ્રેક પડશે, લંચ બ્રેક, એમાં પણ તમે કોઈને 1-to-1 ઓપનલી બોલવાની પ્રશ્ન હિંમત ના થતી હોય તો તમે ગમે ત્યારે એપ્રોચ કરી શકો છો. પ્લીઝ. હેલો. Right. Definitely now government has started certain policies for the carcass disposables. Previously there are open burning sites. Now with the help of, and even how to dispose of those carcasses with the scientific matters, there is a policies are now coming up with the government, because this is the major issues, certain areas where carcasses are disposed of. On the other hand, when there is a large casualties are there, I will give, there are certain biotechnological prepared solutions are there. I will give some content like bioculum. Okay, this is the bioculum is the culture of the bacteria. If any carcass is fall down in the there, they decompose the carcass within 1 to 2 days. So this is the bacteria would generally responsible. Now there is a ready preparations of this bioculum is available. On the other hand, when there is any carcass are disposed at any sites, there is a off smell. બરાબર, જ્યાં ગામમાં આવા બધા મરેલા પ્રાણીઓનો ઢગલો થતો હોય ત્યાંથી નીકળો તો તમને ગંદી સ્મેલ આવે. There is another products are available in the commercial markets. This is sanitrate. These are the bacterial inoculums identified with the help of the biotechnology interventions. Once you spray this sanitrate on the carcass, this bacteria degrades in a way you not get the off smell from the carcasses while it is degraded. So biotechnology and other interventions is already ongoing on. Even in city now sanitariums for the animals are also started. Other essence. That's why we said the One Health approaches. We have to maintain the all ecologies. Okay, that's why there are certain at the village level there are sites we are not restricted, but in the city level they have now started. Thank you. Or for the vulture harvestings, once it was finding out certain medications like diclofenac, sodiums and other antibiotics, Department of Animal Husbandry has already banned diclofenac uses since last many years in animals. Similarly, many antibiotics are there. It was resistance, and it is very vital for saving the lives, because when any human get the infections and multi-drug resistance are there, there is a choline sulfate. It's a very good drug of choice in the human for the RAS infection treatment. So animal husbandry department has a ban use of cholesterol sulfates for the sake of human health. So if haphazard use of cholesterol sulfate in the veterinary sector, there may be chance of raising antimicrobial resistance. Thank you. Any other questions?
हेलो नमस्कार सबको आई एम अ स्टूडेंट टीवाई माइक्रोबायोलॉजी वीएनएसयू सूरत से. मेरा क्वेश्चन ये है कि हम इधर वाइल्ड लाइफ कंजर्वेशन मैनेजमेंट और सबकी बात करते हैं. क्लाइमेट चेंज सबसे बड़ा इश्यू है, एक हॉट टॉपिक है पूरे वर्ल्ड के लिए. वो ना ही वाइल्ड लाइफ को भी अफेक्ट करता है, ह्यूमन लाइफ को भी, और डिजीज कॉजिंग का एक फैक्टर क्लाइमेट चेंज भी है. तो बायोटेक्नोलॉजी इस एस्पेक्ट में क्या कर सकते, क्लाइमेट चेंज को हम क्या हमारा के इन्वॉल्व, बायोटेक का क्या इन्वॉल्व कि क्लाइमेट चेंज में क्या रोल है?
Exactly, climate change. Our visionary Honorable Prime Minister Narendra Modi sir have established like GSBTM, and this is the first state, our department, where have they developed the climate change department. Gujarat is the first state in the country having a climate change department. There is a different portfolios are there. Now come to the questions, how climate change can be changed with the ecological harmonizations. Okay, why this climate change is happening? Due to the ecology imbalances, these climate changes are happening. Here we are addressing bioresilience. What means of the bioresilience? Survival of the all our ecology system. If we are arsening our ecology, the climate will be disabled. Not only you have said human and wildlife, it's also significantly affect the livestock. અત્યારે વરસાદ પડ્યો, મગફળીનો પાલો ક્યાંય મળતો નથી, જે સૌરાષ્ટ્રની મેઇન પ્રોબ્લેમ છે. યુ કેન અન્ડરસ્ટેન્ડ, મગફળીનો પાલો બરાબર એનિમલનો મેજર ખોરાક હતો, એની ઉપર વરસાદ પડ્યો બરાબર છે, તો ફંગસ મોટા ભાગે થઈ ગયું. તો ધીસ આર નોટ ઓનલી ઇફેક્ટેડ, ધીસ ક્લાઇમેટ ચેન્જ ઇઝ નોટ અફેક્ટેડ farmers who are growing the crops, all ecosystems કરે છે. તો ઇકોસિસ્ટમ્સને આપણે જેટલી બેલેન્સ રાખીશું, પોલ્યુશન્સને આપણે ડાઉન કરીશું બરાબર. નોટ ઓનલી વન સેક્ટર કેન ડુ, ઓનલી બાયોટેકનોલોજી કેન નોટ ઇન્ટરવેન્ટ વિથ ધ ક્લાઇમેટ ચેન્જ, but other departments like pollution control departments are there, industrial control departments are there. We are now shifting diesel car to the petrol car and petrol car to the battery operated car. These are the small steps will help to control this climate change. Thank you. Okay. Thank you.
Thank you so much. And now it's time for our next speaker to come on stage and deliver their technical session, and that is Dr. Nishid Dhareya, Director, Center of Excellence for Wildlife, BKNMU. And he'll be leading on applications of biotechnology in wildlife research. So let us all put our hands together.
Good morning, everyone. I think I'm audible to all. Thank you. So I'm here to represent our Center of Excellence for Wildlife and Conservation Studies. And I'm just here to talk about how wildlife conservation can be advanced using the biotechnology. So I am going to talk about what are the areas where we can apply the biotechnology, and I am also going to discuss some of the ongoing projects, some of the completed projects of of wildlife in which we have applied the biotechnology. So we all are aware about the 3 levels of biodiversity. Out of these 3 levels, the one level is genetic diversity, where we are studying the diversity at the gene level. And all our conventional research on wildlife is either the ecological research, where we study the ecology of the entire population, or ecology of a single species, or ecology of a single individual. Sometimes we are studying the distribution of the species. Again, most of the field work and field research is included in that. We study the behavioral study of the animal behavior. We do population study and phytosociology, and we are also doing taxonomic research. So these are all the conventional wildlife research that we used to carry out in the field as well as in the lab. But if we want to inculcate some advancement using the biotechnology, so we can do it with the ecological modeling, where we can use the important software, advanced level software, remote sensing and GIS in that. We can do genetics, we can use genetics in the wildlife. We can apply microbiology in the wildlife, forensic science or wildlife forensics, and the disease diagnostic in the wildlife. Even the hybridization in the wildlife, because nowadays this hybridization is a very big issue in the wildlife, when the wildlife is living in a close connection with the human population. Most importantly, what we need to study the wildlife when we use biotechnology is the sample source, and sample source for the from the wildlife is the DNA source. So DNA source we can take from the invasive and the non-invasive samples, and mostly we used to have non-invasive samples where we do not harm the wildlife, because most of the wildlife is protected under the Wildlife Protection Act, and you cannot harm the animal, you cannot touch the animal, you cannot take blood or tissue out of the animal, and that is why we are mostly depending on the non-invasive sample. So non-invasive samples are the samples which we collect from the wildlife without harming them. So we used to collect the scat of the animal, the hair of the animal. Sometimes we found the dead animal in the jungle, dead animal due to the road collision. So at that time we are collecting the samples from the wildlife, and this is how we collect the sample, and those collected sample we can send out to the biotechnology laboratory. Now these samples will help us to know the species, because many a time there are varieties in the species, there are variety in morphology, but genetically the species is the same. So if we want to identify the species at the gene level, DNA fingerprinting, DNA barcoding, that help us. Most of the time what happens in the field research, in the conventional wildlife research, when we are identifying the species by just a scat. So the scat of jackal and scat of dog are very same. Now jackal used to come to the village and dog used to go to the forest, and when they put dropping in the forest and we collect it, we thought it is a dog, we thought it is a jackal scat or it is a hyena scat, because the shape, size and color are almost same. So in that cases, if we do the DNA analysis, we can easily know whether it is a dog, domestic dog or jackal. Similarly, we have jungle cat in the jungle, we have rusty spotted cat in the jungle, and we have domestic cat in the villages. And there is a chances of hybridization, there is a chance that we need to identify whether this particular hare or this particular cat is a domestic cat or it's a jungle cat or it is a what kind of cat. In that cases, the species identification using DNA is very helpful. So this is how we can inculcate, we can apply biotechnology while we are doing the field research in the wildlife. The another excellent example is tiger census. We all know that all India tiger census is going on, and we are identifying tigers by their stripe pattern, individual tiger. Similarly, we are identifying individual lion by the whisker spot pattern, just by the morphology. But what about my sloth bear? I am a sloth bear biologist, I am a bear biologist, and sloth bear, you can see the sloth bear is entirely black. There is no pattern, there is no design, there is no marking on the individual sloth bear. Now, if you want to identify the individual sloth bear, you have the only option is the DNA. So, if you can collect the hair samples, if you collect the scat samples, then we can get the exact. Today, we have the exact number of lion, we have the exact number of leopard, we have very precise number of tigers in our country, but we do not have the precise number of sloth bear, and the reason is we cannot identify the individual sloth bear. So, this is how the biotechnology is helpful. Then the very important issue nowadays, it is addressing the health issues, wildlife health issues and diagnostic, because wildlife population is increasing day by day because of the successful conservation efforts of the forest department, because our own ethics towards the wildlife, our culture, Indian culture towards the wildlife, we are successfully we are success in increasing the number of wildlife. So, wildlife population is increasing, but on the other hand, the locations, the place for the wildlife is decreasing, or it is as stable as it is. So, the wildlife has started moving towards the human dominated area, towards the farm, towards the villages, and this is what zoonosis can be start. So, disease which come from the wildlife or disease in wildlife that can come from the humans. So, that zoonosis process is going on. So, for that we have tried to study such wildlife which is living very close to the human population, and this is none of the one, it is a sloth bear. If you go to Mount Abu, my friends, in Mount Abu you can see sloth bear on the road, you can see sloth bear in the hotel, you can also see sloth bear moving in your resort, and if you go out at 11 o'clock, 12 o'clock in the market of Mount Abu, you will see sloth bear eating the garbage. Sloth bear is also eating the prasad from the temple, the offerings to the god, and this is all chances by which the human pathogens or the microorganism from human that can come into the sloth bear. So, you can see here the sloth bear eating the garbage, and this is a big issue in Mount Abu, that sloth bear are now started eating garbage in the Mount Abu. So, what we have done, we have surveyed such areas where there are more number of sloth bear visiting the human dominated areas. We see that in this area sloth bear and human coexist together. There are chances that human are attacked by the sloth bear, sometimes sloth bear is also attacked by the human. So, we selected such area, the one is Jessore Wildlife Sanctuary, and from Jessore Wildlife Sanctuary we visited the edges of the sanctuary કે જ્યાં આગળ માણસો પણ હોય, રહેતા હોય, કામ કરતા હોય અને રીંછ પણ ત્યાં જતા હોય, and we collected the scat of sloth bear from these area, and then we have done the routine microbiological analysis. We have isolated the microorganisms, we have isolated the microbial colony, and we want to identify the human pathogens from those scat, and that is why we use a machine which is called Biotech2 compact machine that can only identify the human pathogen, whether it is a human sample or it is an animal sample. And using the Biotech2 we have generated the reports that what kind of human pathogens are found in the sloth bear gut, in the sloth bear scat, and then we compared those animals, those sloth bear which are in the wild and those sloth bear which are living with the humans in the zoo, and we were surprised to know that we found 11 number of colonies, 19 colonies of human pathogens from the wild sloth bear and 29 colonies of human pathogen from the sloth bear which are living in the captivity. Candida was the fungi which is also found from the wild sloth bear, and it shows that there is a very close connection of sloth bear and the human. And then we tried to use the antibiotic resistance. We want to see whether these organisms are resistant to antibiotic, because these sloth bear or these animals, when they are getting sick, when we are giving them medicine, whether these medicine are good for the animal or not, whether the medicine will be effective. If the organisms are resistant, medicine will not affect. આપણે કેટલી પણ દવા આપીએ એ દવાની અસર નહીં થાય, બિકોઝ ત્યાંના ઓર્ગેનિઝમ છે રેઝિસ્ટન્ટ હશે. સો વી ફાઉન્ડ ધેટ 3 of such organisms in wild sloth bear were resistant. અને ઝૂની અંદર તો 10 એવા ઓર્ગેનિઝમ છે કે જેને કોઈ એન્ટીબાયોટિક અસર નહોતી કરતી. So we can try the same research with the other animal, and we can try to give this message to the zoo. આપણી પાસે ઘણા બધા ઝૂ છે, કેવડિયા ઝૂ, સકરબાગ ઝૂ, બીજા કોર્પોરેશન ઓન્ડ ઝૂઝ છે, અને ત્યાં ઘણા બધા ઝૂ એનિમલ્સ છે. તો આવા રિસર્ચ કરી અને આપણે ઝૂને કહી શકીએ કે તમે આ બધી દવા આપો છો, પણ એ દવા કદાચ તમારા એનિમલને ઇફેક્ટિવ નહીં થાય, બિકોઝ દે હેવ ધી રેઝિસ્ટન્ટ ઓર્ગેનિઝમ ઇન ધેર ગટ. So we found 13 human pathogen from the sloth bear. This is just an example of sloth bear, but similar research we can enhance for all kind of wild animals. 7 were common in between both wild and captive. 6 were opportunistic. 15% of the bacteria from the wild sloth bear were resistant to antibiotic, and in case of captive sloth bear, 53% of the bacteria were resistant to the. And the same research we published in a very reputed journal called Wildlife Biology. Very recently this research was published, and when this research published, it has given us more enthusiasm. આનાથી અમને એક નવું રિસર્ચ કરવાનું મન થયું, and that research is, let us study the diversity of bacteria in the gut of the animal. And then we started 2 such research. One is on lion, to study the gut bacteria of lion, and similarly to study the gut microbial diversity of sloth bear. And again we tried to compare what is the difference between the gut microbial diversity of sloth bear living in the forest and sloth bear living in the zoo. Again we selected 2 areas, collected the sample, but now our aim was not study the human pathogen. Here our aim is to study the entire gut microbial diversity, and that is why again we have to take the help from the biotechnology. So we have used the routine biotechnological method. We sent our sample to the biotechnology lab, where we have done the sequencing of the sample, and we try to identify the bacteria. So you can see these many bacteria where we were found from the captive sloth bear, and out of these bacteria, mainly Firmicutes bacteria were taking part. It was the major role. Now if many of you, if you don't know about sloth bear are myrmecophagous animal. They eat termites, they eat ants, they eat fruits, and they eat honey. બીજું કાંઈ નથી ખાતા, એ માણસો નથી ખાતા, બીજા કોઈ પ્રાણી નથી ખાતા, because sloth bear doesn't have 2 incisor. આ બે આગળના દાંત એને નથી હોતા. So they cannot cut the food, they can suck the food. And they eat entire termite, they eat colony of the termite, they eat colony of the ant. So another question that is again that needs the answer from the biotechnology is, termite is having different toxins in their body, and termite entirely goes into the body of sloth bear, into the gut of sloth bear. તો sloth bear ના ગટમાં એવા કયા ઓર્ગેનિઝમ છે કે જે ટર્માઇટના ટોક્સિનને ઓછું કરી દે છે? ટર્માઇટની અંદર જે ઝેર છે, ઉધઈની અંદર જે ટોક્સિન છે, એન્ટ્સની અંદર જે ટોક્સિન છે, એ ટોક્સિન ડિગ્રેડિંગ ઓર્ગેનિઝમ ત્યાં હોવા જોઈએ. So that research we have started, and for that we need to know the entire gut diversity of the sloth bear. So this research is going on as a PhD research at our center, and 2 students, they are studying the gut microbial diversity in the sloth bear. So this is what found in the wild sloth bear. Now how it helps to the conservation of the wildlife. So we found 3 major phyla, and we generally found these 3 phylum of bacteria from every animal. This is Firmicute, bacteroids, and proteobacteria, and the balance of these 3 phylum of the bacteria in the gut of animal is very required. એની હેલ્થ માટે આ ત્રણેયનું રેશિયો બહુ જરૂરી છે. સવારે વીસી સાહેબે કીધું ને કે ટીચર સ્ટુડન્ટનો રેશિયો 1:15, 1:30, તો આ રેશિયો છે બહુ જરૂરી છે મેન્ટેન થવો. એવી રીતે આ જે 3 ગ્રુપના બેક્ટેરિયા છે એનું આપણા પેટમાં એનો રેશિયો મેન્ટેન થવો ખૂબ જરૂરી છે. So we try to study the ratio of these 3 bacteria which is responsible for the health of the host. પછી એ sloth bear હોય, lion હોય, rhino હોય કે elephant હોય, બધાના ગટની અંદર આ 3 ગ્રુપના બેક્ટેરિયા હશે. So the FB ratio, firmicutes and bacteroid ratio in the gut of animal is very important. So here this is the ideal ratio of firmicutes bacteria and bacteroids bacteria is 0.57. This is the ideal. Now if we study any animal using the biotechnology, using the wildlife research in any forest, say for example if we study leopard and if we found the FB ratio in the leopard gut which is higher or lesser than 0.57, then we can say that there is some dysbiosis, there is some health problem in the population, and immediately we can inform to the forest department or forest manager that this particular population of wildlife is having some issue, and then we can do further research to find out what are the issues with the animal. So this is how we can study or we can do wildlife management and wildlife conservation using the biotechnological research. So again, what we found that common phylum between the captive and the wild sloth bear, captive and the wild sloth bear, number of classes, how many common, how many unique, and how many are common between these two. Similarly, biotechnology can help to understand the structure of the population. Now again, this is very important, population structure. How many male, how many female, how many dependent cub, કયું કબ છે એ કયા પેયરમાંથી આવ્યું છે, because inbreeding is going on. એક genetic bottleneck. If you know about lion, if you consider the lion population before the independence, there were only 60 to 100 lions, and if we consider today's lion population, it is more than 800 lions. So આ જે પણ 800 lion છે, the entire population is passed through a bottleneck. That means the genetic makeup of this entire lion population is almost similar. So if you want to study the structure of this population, again your conventional wildlife research or going in the field, looking at the animal, recording the data, it may not help. You need to know the genetic structure of the population, and for that you need to have the help of biotechnology. So similar research we can study because our forest is now fragmented. There are very small patches of the forest in entire Gujarat state, and Gujarat state is very seriously, Gujarat State Forest Department, they are very seriously looking after connecting the habitat, which is the project which is known as connecting dots. Forest હવે dots જેવા છે એને connect કરવાનું કામ શરૂ કર્યું છે, which is also known as ecological corridor. So whether the wildlife are using the ecological corridor or not, અહીંયા આગળ મેં એનિમલ જોયું અને અહીંયા આગળ એનિમલ જોયું તો આ એનિમલ અહીંયાથી આવ્યું હશે કે નહીં એ મારે જાણવું છે. તો એના માટે again what we require whether the animal was same or not, અને જ્યારે એ જોવું હશે individual same છે કે નહીં, at that time we need to have the DNA of that animal. So we are doing the similar project in Melghat in Madhya Pradesh, Maharashtra, where we are trying to identify whether the corridors are used by the bears. So what we are doing, we are collecting the bears scat samples from bear from entire Melghat region, and you can see here all these red dots are the sloth bear scat samples. હવે આ sloth bear scat sample મળ્યા, that means that sloth bear is using the corridor, but whether the corridor are used by the same individual, અહીંયાનું individual અહીંયા ગયું હતું કે નહીં એ જોવું હશે. So again what we need to do, we need to do the DNA analysis, we need to know how the gene is flowing, how the animal is using the corridor. So that project we have done in Melghat. It has given very promising result, and we have told Maharashtra Forest Department that your Melghat corridor is active for the sloth bear, and now Maharashtra Department has started conserving and protecting that corridor. So similar module we have suggested to Gujarat Forest Department, because in Gujarat also there are good ecological corridors for lion, leopard and sloth bear, and we are going to start this project in Gujarat also, where we study the joining the dot project, where we do the DNA analysis of the scat collected from lion, leopard and sloth bear, and you will see that how the corridors are used and whether the corridors is really required or not. So, this kind of projects we can start, this kind of research we can do, and whatever conventional research we are doing using our field observation, we can inculcate the knowledge of biotechnology, we can take help from the biotechnology, and we can do the project in such a manner that biotechnology can give the robustness to our observation and our conclusion. So, it is very important that we start using the advanced technology. આપણી પાસે પેલા સ્વિચ વાળો મોબાઈલ હતો અને સ્માર્ટફોન આવી ગયો અને we happily accepted that smartphone. Similarly, initially we were doing conventional wildlife research by going out in the field, collecting the photograph data and old field data. But now when the advancement is available with us, why don't we use the advanced method? હવે આપણે advanced method નો ઉપયોગ શું કામ ના કરવો જોઈએ? પહેલા આપણે ખાલી એનિમલ ને જોઈને આપણે સ્પીસીસ આઈડેન્ટિફાઈ કરતા હતા કે ઓકે પીઠ ઉપર આટલા ભીંગડા છે, અહીંયા આ છે, અહીંયા આવું માર્કિંગ છે તો આ સ્પીસીસ છે. Because at that time we were not having the advancement. પણ હવે તો આપણી પાસે DNA fingerprinting છે. We have the barcodes animal, barcoded plants and animal. So now we should use those barcoded so we can give more robust data and more robust identification of the species at the gene level also. And our identification would remain more authentic using the biotechnology. So this is how we can use the biotechnology in the wildlife research. I think I just delivered what I was having in my cup and in my ball, and what kind of research we are doing at the Center of Excellence. And I invite you all to join Center of Excellence for Wildlife Research and Conservation Studies, where we are going to have the advanced level research lab where we can do the biotechnology experiments. We have the advanced level GIS lab where we can inculcate the advancement in the wildlife conservation, and you can do your research. We can help you. Our team of scientists and researchers can also help you to do your research or your internship from this. So with this, I thank you all and thank you very much. Namaskar. And I would be happy if you have any questions or any suggestions, I would be happy to discuss with you. Yes.
Hello. So, as you mentioned that the exact number of sloth bears are not known. So, could we use tagging as a source of method for estimating the population?
For sloth bears?
For sloth bears.
Sloth bears, right now, all the forest department of entire India, because 85% population of sloth bears is in India. Sloth bears are found only 3 countries, Nepal, Sri Lanka and India. Out of these 3, we are holding 85% share. So everywhere sloth bear population census is done by the conventional method, that they sit on the full moon night, they sit at the water hole and they count the head of sloth bear, head numbers of the sloth bear. So that will give only guesstimate, not the estimate. So this is why we can say the exact number of sloth bear is not known.
But sir, tagging?
Tagging we can do, but again for that we need to have a permission. And how many sloth bear you will tag?
But sir, only through tagging we can know the exact number.
More easy method is just to have the DNA sample, collect the scat, identify the individual, and you can say this forest area is having 4 sloth bear or 5 sloth bear. So chances of repetition would be very less.
Thank you, sir.
Yeah, thank you. Yes, one more from here.
Namaste, sir.
Namaskar.
Sir, what is the status of extending area in future and now by sloth bear? Is it like lion extending area up to one district to 11 districts.
Yes.
And another one is what type of the myrmecology, myrmecophagus is used by sloth bear is an opportunistic thing. But a special ant have been selected for food, or any research have been done on the sloth bear for the special ant those are used for
Specific species of ants. Yes, this is a very good question, because there are several species of ants and termites are available, and we actually need to know that whether sloth bear is choosy like us, like આપણી પાસે dairy milk પણ છે, KitKat પણ છે, પણ આપણે dairy milk જ ખાઈએ છીએ કે eclairs ખાઈએ છીએ. So several species of ants. Again, it is very important to know what kind of ant species are available, what kind of ant species we found from the scat of the sloth bear. And when we know that, we can identify that sloth bear is preferring only these particular ants or these particular species of termites. So such research is not still carried out, but there is a need to do this research also. Yes, sir. One student from there. Yeah.
We don't get the data to perform in lab. For the lab work also, we need the data
From the field.
according to traditional ecology.
From the field, yes.
So I wanted to know about the traditional approach and biotechnology in the future. Means what are the scope and what are the approaches to that?
Yeah, I would say it's a combination of conventional approach and the advancement. We cannot leave the conventional approach, because if you want data from the wildlife, you have to go to the forest. But after coming back from the forest, you have an entire lab of biotechnology. So why don't you use your samples, why don't you use biotechnology there? So you can reach up to the gene level, up to the genetic level of your sample. So your data would be more authentic and your conclusion would be more strong to stand at the world level.
Thank you, sir.
Yeah. I think, thank you very much. Thank you all.
Thank you so much, sir. And before we proceed ahead with the next speaker, here I would like to honor our Shri MLA, Shri Sanjay Koradia ji, who has gave his gracious presence among us, and hence I would request Digvijay sir, Shri Digvijay sir, to kindly felicitate him on the stage, please. Also, I request Shri Sanjay sir to kindly share a few words. Sir, તમારા બે શબ્દો please.
ગુજરાત સરકાર દ્વારા રિસર્ચ માટે અને એ ખાસ કરીને વાઇલ્ડ લાઈફ એમનો રાજ્ય કક્ષાના કાર્યક્રમો થતા હોય છે, અને આ લગભગ મને એવું લાગે છે કે આ કાર્યક્રમ ડીડી જાડેજા સાહેબ એટલે અમારા પહેલાં અહીંયા જૂનાગઢ માટે એટલા માટે ગૌરવ છે કે જૂનાગઢના અહીંયા એસડીએમ તરીકે સેવા આપી હતી ને અત્યારે આ વિભાગના ડાયરેક્ટર તરીકેની એમની જવાબદારી છે ને એવું જૂનાગઢની અંદર એક આ કાર્યક્રમ એટલે અમારું આ નરેન્દ્રભાઈના શબ્દમાં કહી તો આ દેશનું ભાવિ એવા યુવાનોની સાથે આજનો આ વાઇલ્ડ લાઈફનો સંવાદ. આમ જોઈએ તો ખૂબ ખૂબ અભિનંદન આપું છું જાડેજા સાહેબ અને ગુજરાત સરકારને કે આ મારા મત વિસ્તારની અંદર અમારા બાળકોને આનું નોલેજ મળે. અને ઘણી વખત આપણે એવું થતું હોય છે કે આ વિષયની અંદર શું છે સાહેબ, આજે તો એક સંયોગ ભેગો થઈ ગયો છે. હમણાં એક ફંક્શનમાં હતો ને ત્યાં અમેરિકન વ્યક્તિ હતા, એમની સાથે હમણાં કલાક પહેલાં જ ચર્ચા થઈ, એટલે એમ કહેતા હતા કે રિસર્ચની અંદર આપણે હજી ઘણા પાછળ છીએ કે જે બજેટ રિસર્ચમાં થવું જોઈએ. કારણ કે સૌથી મોટું પાસું એ છે કે એની પેટર્ન બને, એની અંદરથી જે તકલીફો હોય એ દૂર થાય અને ભવિષ્યની અંદર કંઈ રોગો ના આવે. આપણે ઘણી વખત એવું છે ઘણી પ્રજાતિઓ જે આપણે જોઈએ છીએ કે ભૂતકાળની અંદર અમુક રોગોને હિસાબે અત્યારે પ્રજાતિઓ રહી નથી, તો એની અંદરથી કઈ રીતે આપણે વાઇલ્ડ લાઈફને બચાવી શકીએ જનરલી, તો એમના માટેના રિસર્ચ માટેની આખી આ સબ્જેક્ટ છે. અને મને તો સાહેબ હું પહેલી વખત હમણાં ધારાસભ્ય બન્યો ને પછી જંગલમાં આરએફઓ સાથે હું ગયો હતો ચક્કર લગાવવા, ત્યારે અંદર દીકરીઓ 5-6 હતી. એટલે મને કે રિસર્ચ કરે છે પતંગિયા ઉપર આખી અંદર જંગલની અંદર. એટલે એમની સાથે વાત કરી એટલે આપણે એવો વિચાર થાય કે માણસની અંદર પતંગિયું એના વિશે તમે શું વિચારી શકો, પણ એમના વિશે પણ દીકરીઓ, આ દેશની આ રાજ્યની દીકરીઓ રિસર્ચ પતંગિયા વિશે કરતી હોય છે. તો એના માટેનો આ ખૂબ મોટો એક સબ્જેક્ટ છે. એટલે અહીંયા દીકરા દીકરીઓ બેઠા છે, મારે તો એમને ખાસ એટલા માટે કહેવું છે કે રૂટિન પ્રક્રિયા એ તો જીવનનું પ્રણાલી છે. સવારે ઉઠવું, ફ્રેશ થઈ નાસ્તો કરી કામ કરવું, બપોરે જમવું, પાછું રાજકોટ જેવી આદત હોય તો સૂઈ જવું 2 કલાક 2 થી 4, અને પછી પાછું સાંજે કરવું. એ રૂટિન પ્રક્રિયા છે. પણ એનાથી કંઈક નવો વિચાર તમારી અંદર આવે કે કંઈક માઇન્ડની અંદર એવું કન્સેપ્ટ ચેન્જ થાય કે આપણે આપણા પોતાના મગજથી આપણા વિચારો, આપણી શક્તિ અને એમની સાથે આપણે કંઈક દેશને અને દુનિયાને આપી શકી એ જ માણસ છે. કારણ કે રૂટિન પ્રક્રિયામાં કોઈ નથી. તમે નવું કરશો તો લોકો તમારી સામે જોશે, તમારો વિચારો નવા આવશે કે તમારી અંદર નવી શક્તિ આવશે ને તો એ શક્તિનો કોઈક પૂજા કરશે. આ દેશની અંદર, દુનિયાની અંદર તમારા વિચારની, તમારા નવા વિચારની શક્તિ છે. રૂટિન પ્રક્રિયાની કોઈ વેલ્યુ નથી. તમે જે કામ કરો છો એ એક થઈ ગયું છે. અને આપણે બધાને ખબર છે કે આમાં અત્યારે એમાં ઘણા તમે બેઠા છો, એક બહાર નીકળી અને એક લેવલે તમે જોબ કરશો. જોબ તમારી 10,000થી લઈને 10 લાખ સુધીની હશે. એ જોબની અંદર તમે શું નવું ઇનોવેશન કરી શકો છો, તમારો આઈડિયા ઇનોવેટિવ કયો છે, એની અંદર તમે શું કરી શકો છો, એ સૌથી મહત્વનું પાસું છે. એટલે મારે બધાને વિનંતી છે કે તમે આજે આ કન્સેપ્ટની અંદર, નહીંતર માણસને એવું છે કે ભાઈ અત્યારે એજ્યુકેશનની અંદર એવું થઈ ગયું કે ડૉક્ટર ને એન્જિનિયર એ સિવાય કંઈ છે જ નહીં. એ મગજમાંથી કાઢી નાખવાનો સમય છે. સારો દરજી પણ દુનિયાની અંદર સારું ફેશન ડિઝાઇનિંગ કરી અને કરી શકે છે. સારો મોચી સારા ચપ્પલ બનાવી શકે છે. એટલે આ બધી વસ્તુ છે. એટલે એવો મગજમાંથી કાઢી નાખવાનું ડૉક્ટર એન્જિનિયર છે એટલે દુનિયા છે. તમારા મનની અંદર નવો વિચાર છે તો દુનિયા છે. એ વિચાર છે એ વાઇલ્ડ લાઈફનો વિચાર આવ્યો ને એ મહત્વનું છે. એટલે આજના સેમિનારની અંદર તમે પાર્ટિસિપન્ટ લીધું છે એટલે તમને બધાને ખૂબ ખૂબ શુભેચ્છાઓ. અને તમારા વિચારો પણ અમને આપો. તમારા આઈડિયા, તમને એવું લાગે કે આવું કરવું જોઈએ સરકારે. એવું નથી કે અહીંયા આ ફર્સ્ટ રોમાં બેઠા છે અધિકારીઓ કે અમને બધાને ખબર છે. અમારા કરતાં તમારી પાસે શક્તિ વધારે છે, તમારી પાસે વિચારો વધારે છે. કારણ કે ઘણી વખત કહે છે ને વડીલો કે કેટલી દિવાળી જોઈ, અમે કંઈ કે આપને કહે છે કે તમારા કરતાં જાજી દિવાળી જોઈ, પણ આપણે એને કહી શકીએ કે ફટાકડા અમે ફોડ્યા છે. બરોબર છે ને? દિવાળી જોઈ મહત્વ નથી, ફટાકડા ફોડવા માટેની દિવાળી છે, દીપ જલાવવા માટેની દિવાળી છે, તો એ મહત્વનું છે. એટલે અમે બેઠા છીએ એટલે અમને બધી ખબર છે નહીં. તમારી પાસે પણ શક્તિ છે. તમારા વિચારો સમયાંતરે આપો, અમને આપો, જે તે ડિપાર્ટમેન્ટ હોય એમને આપો કે આવું કરવું જોઈએ. બધું થઈ જવાનું એવું શક્ય નથી, પણ એમાંથી એક વિચાર નવો દેશ અને દુનિયાને બદલવા માટે ખૂબ મહત્વનો હોય છે. એટલે એ પાસાની અંદર તમે બધા આગળ વધો અને જ્યારે પણ જે જરૂરિયાત હોય ત્યારે 100% અમને પણ કહો અને અધિકારીને પણ કહો. સરકાર, નરેન્દ્રભાઈના વિચાર સાથે સરકાર, ભૂપેન્દ્રભાઈની શક્તિ સાથેની આ એક શક્તિ એ આપણા જીવનની અંદર એક નવું પરિવર્તન આવે ને આપણે એક રાજ્ય અને આ રાષ્ટ્ર એ ભારત માતા પરમ વૈભવ સુધી કહીશું કે પહોંચી શકે. ખાલી મનુષ્ય નહીં પણ પશુ જીવ, પક્ષી જીવ છે એને પણ આપણે એમના માટે, જીવન માટે એમને તક આપી શકીએ, એમના પ્રયત્નો આપણે કરી શકીએ, એ સૌથી મહત્વનું છે. ફરીથી આજનું ડીડી જાડેજા સાહેબ અને સમગ્ર ટીમને જૂનાગઢમાં આ કાર્યક્રમ કરવા માટે ખૂબ ખૂબ શુભકામનાઓ. અને તમે બધાએ જે પાર્ટિસિપન્ટ લીધું છે તમને પણ ખૂબ ખૂબ શુભકામનાઓ. આભાર. વંદે માતરમ. ભારત માતા કી જય. જય જય ગરવી ગુજરાત.
Thank you so very much, sir. Thank you so much. Aabhar. And now I would like to honor and felicitate our City BJP President, Shri Gaurav Ruperelia, and for which I request Dr. Snehal Bagatharia to come forward and do the honors. And once again, continuing with the session, technical session, and here it's time for me to call upon stage Sri Raghuveer Singh D. Jadeja, ex-forest officer, and he'll be taking session on evolution and conservation genetics. I would request our speakers and our technical session leaders to kindly make it crisp between 10 to 15 minutes for all of us. So once again, let me call upon stage Sri Raghuveer Singh Jadeja ji. Let us all put our hands together for him.
ખૂબ ખૂબ ધન્યવાદ. જય હિન્દ, જય ભારત. અમારે સેશન તો આવતીકાલે છે. ખરેખર હું ફોરેસ્ટ ઓફિસર છું, પણ બીએસસી એમએસસી એગ્રી મેં અહીંથી પૂરું કર્યું છે, જુનાગઢથી. અને પેલો ફોરેસ્ટ ઓફિસર છું કે નોકરી ભણતર પૂરું થયું અને તરત આ ગિરનારની અંદર મારી પહેલું પોસ્ટિંગ હતું. અને ડીડી જાડેજા સાહેબનું પણ આ વિસ્તારની અંદર પોસ્ટિંગ, જ્યાં તમારું પોસ્ટિંગ થાય છે ત્યાં પોતીકું ઘર બની જતું હોય છે. એટલે હંમેશા બીજે ગમે ત્યાં નોકરી કરીને આવો, પણ છતાં લગાવ હંમેશા આપણે જ્યાં સારી નોકરી કરીએ, જ્યાં પહેલું પોસ્ટિંગ થયું ત્યાં થતું હોય છે. સંજય કોરડિયા સાહેબ આજે મળવાનો મોકો મળ્યો, અને એક આરએફઓ સાથે તેઓ જંગલમાં એમ કે અમે ગયા તો શું જોવે, નેગેટિવ જોવે. પણ તેમને તેમના આપણા વિસ્તારના જે દીકરીઓ, જે બાળકો કે જે કંઈ શું કરી રહ્યા છે, એ એક પદાધિકારી પૂછવું કે એમાં ઇન્ટરેસ્ટ લેવો, એ આપણા જે યંગ જે છોકરાઓ છે એને ખરેખર ખૂબ આગળ રસ્તો ચીંધી શકે છે. તમે ખાલી એટલું પૂછો કે નરેન્દ્ર મોદી સાહેબ ઘણી વખત કંઈક એક ચોકીદારને પણ પૂછી લેતા હોય છે કે ભાઈ દીકરા શું કરે છે, તો તમે એને તમે પદાધિકારી, કારણ કે તમને બધા જોવે છે, તમને અનુકરે છે, તમને અનુસરે છે બધા, એ તમારો એક શબ્દ આપણા ઘણા બાળકોની જિંદગી ફેરવી દેતો હોય છે, ઘણી જગ્યાએ આગળ વધતા હોય છે. મારો જો રેકોર્ડ તમે જુઓ ને સાહેબ, તો મારી 41 વર્ષની ફોરેસ્ટની સર્વિસની અંદર 8,500 સાપો મેં પકડ્યા છે. કેટલા? 8,500. બીજો એક મારો એક રેકોર્ડ છે સાહેબ. ઘણા માણસો એમ કે ભાઈ આમને દબંગ ઓફિસર આટલા આતંકવાદી માર્યા ને ફલાણું ના, કારણ કે હું પાછો સાહેબ મિલિટરીનો માણસ છું, આસિસ્ટન્ટ કમાન્ડન્ટ હતો, અને ત્યારે ભારત દેશ બહુ સળગી રહ્યો હતો, જ્યાં જુઓ ને આ બોમ્બ ધડાકા. ત્યારે સાહેબ ગાડીની અંદર તમે રેલવે મુસાફરી, અહીંયા તમે બેઠા હોય તો અજાણી વસ્તુને ટચ કરવાની સાહેબ મનાઈ હતી, પણ અત્યારે એટલી બધી શાંતિ છે, બોમ્બ ધડાકો શું આમ ફટાકીઓ ન ફૂટે એટલી બધી સલામતી છે. પણ આપણે એક ફેજ એવો પસાર કર્યો હતો જે હું જ્યારે આમાં હતો ત્યારે ગાડીમાં સાહેબ બેસી ન શકાય, એટલી બધી અનસિક્યુરિટી. ત્યાર પછી હું જીપીએસસી પાસ કરીને ડાયરેક્ટ ફોરેસ્ટમાં આવ્યો. પણ મારો એક બીજો પણ રેકોર્ડ છે સાહેબ. મારું ફિઝિક તમે જોશો તો ફોજી જેવું લાગે, પણ હજી સાહેબ 59 વર્ષની ઉંમરમાં હજી કદાચ મગર મારા હાથમાંથી છટકી ન શકે સાહેબ. ન કે ખાધી છે માગ્યા ઘરની છાશ, કારણ કે અમારો ઉછેર એવો થયો છે. પણ એક બીજું સાહેબ, હું જ્યાં રિટાયર્ડ થયો ને હમણાં, તો મને જરાક નિરાશાજનક થઈ ગયો. કહો એટલે રિટાયર્ડ થઈ ગયા આપણે, હવે નોકરી પૂરી થઈ ગઈ. પણ મેં રાહુલ ગાંધીને જોયો ને કે 56 વર્ષનો રાજકુમાર, તો મેં કીધું એટલે હું તો હજી એના જેવડો જ છું. એટલે સાહેબ બહુ તમને ઇનડાયરેક્ટલી આવી કોઈને નેગેટિવ પ્રેરણા મળતી હોય છે. પણ સાહેબ મારી મોટાભાગની નોકરી ક્રોકોડાઇલ સાથે ગઈ. એટલે આ સાપ પકડું ને સાહેબ, મને 8,500 સાપ પકડ્યા હશે. પણ એમ કહેવાય છે અમારા કચ્છી સાહિત્યકાર સાથે હું રહ્યો. હું સાહિત્યનો માણસ છું. કચ્છ મિત્રમાં કોલમ લખતો સાહેબ વન્યપ્રાણી સંપદા ઉપર. મારી સાહેબ આ ગુજરાતની ભેટ છે એક પક્ષીની. જે અંગ્રેજો આપણા દેશની અંદર પક્ષી શોધી ગયા ને એવી જ મારી એક ઇન્ડિયન સબકન્ટિનેન્ટની મારી ડિસ્કવરી છે. રૂફસ્ટેડ રોકથ્રસ, એક લાલ પક્ષીની શોધ. મારી એક પક્ષીની રીડિસ્કવરી છે સાહેબ, જેનું નામ છે ફર્નિડા સ્ટોલિસ્કા, જે 1872ની અંદર આપણા ગુજરાતની અંદર એ સાયન્ટિસ્ટ હતા અને પક્ષી શોધીને ગુજરી ગયા સાહેબ. એ પક્ષી કોઈના હાથમાં ન આવ્યું, એ મારા હાથમાં આવી ગયું સાહેબ. ત્યાર પછી મારી એક સાપની નોંધ છે, ઈચી સોચુરેક્સ શોખની. પછી મારી 2 પતંગિયાની નોંધ છે સાહેબ. પતંગિયા કીધા એટલે હું જ્યારે એસઓયુમાં નરેન્દ્ર મોદી સાહેબનો જ્યારે બહુ સારો ટેન્યુર હતો, સારા અધિકારીને એસઓયુમાં એકઠા કરેલા, ત્યારે મારું પણ એમાં નામ આવ્યું કે આને મૂકો તો આપણું સારું દેખાય. તો ત્યાં મૂક્યા ભેગા સરદાર પટેલ મૂર્તિની બરોબર સામે પગથિયાની બાજુમાં વન વિભાગની નર્સરી, ત્યાં હું સાયકલ લઈને ત્યાં ગયો અને એક ફોટો લીધો. એ ફોટો મેં ક્લિક કર્યો તો બધાને ખબર પડી કે આ તો નવું પતંગિયું છે, કમાન્ડર નામનું બટરફ્લાય. એટલે કચ્છી સાહિત્યકારે એમ કીધું છે કે હું 35 ભેગો ઢોર છે સાથે રહ્યો છી, હેજા ઢોર ઢીક નહીં મારતી, સિટીમાં માણસો હાલનું નહીં દીતા. કચ્છી ભાષામાં એમ કહેવાય છે કે 35 વર્ષ હું ઢોર ભેગો રહ્યો છે બાવા. કચ્છમાં જાડેજાઓને બાવા કહે, પણ મને ઢોરે આમ ઢીક મારી નથી. પણ આ નલિયાની બજારમાં માણસ હાલવા દેતા નથી, કારણ કે આપણા કરતાં ઢોરમાં ડિસિપ્લિન વધારે છે. એવા એટલે સાહેબે કીધું કે હંમેશા જાડેજા સાહેબે બાયોટેકનોલોજી વિભાગ, તમને ખબર છે આપણે વૈદિક એરાની અંદર એક બાયોટેકનોલોજીનો એક ઇવેન્ટ બની ગયો છે. કોઈ ભાગવત વાંચ્યું છે રામાયણ મહાભારતમાં અંદર, કે જ્યારે માતા ગાંધારીનો બાળક મૃત્યુ પામ્યું અને એના ગર્ભમાંથી જ્યારે વેદ વ્યાસે જ્યારે 100 બાળકો ઉત્પન્ન કર્યા ને, આ બાયોટેકનોલોજી વેદ કાળથી આપણે વૈદિક બાયોટેકનોલોજીથી ચાલુ કરી અને આજ સુધી. અને અત્યારે આ જાડેજા સાહેબ હું વાત કરતો હતો કે મેં ઘણી બધી વાઇલ્ડ લાઈફની કોન્ફરન્સો આપણે જોઈએ છે, ઘણી બધી સ્ટડી છે, પણ આખા બાયોટેકનોલોજી વિભાગમાં સાહેબ મિશન કમિશનર તરીકે આવ્યા અને બાયોટેકનોલોજીને આગળ વધારવા માટે, કારણ કે આપણે અત્યારે ઘણું ગુમાવી રહ્યા છીએ. આપણે સિંહો ગુમાવી રહ્યા છીએ, વસ્તી વધે છે પણ આપણા કમ્પેરિઝનમાં ગુમાવી રહ્યા છીએ. દીપડાઓની સાથે કન્ફ્રન્ટેશન થઈશું, આપણે રીચ ગુમાવી રહ્યા છીએ સાહેબ. આપણે જો જોવા જાઓ તો હવે વીછી ક્યાં જોવા મળે છે? તમે વીછી જોયો ક્યાંય? ખેતી કરવા જાઓ તો વીછી જોવા મળતા, કારણ કે આપણે ઝેરી દવા છાંટી રહ્યા છીએ. એ બાયોટેકનોલોજી આપણને આવી જે સ્પીસીઝ બચાવવા માટે ઉપયોગમાં આવે. અમે જે વાઇલ્ડ લાઈફના માણસો છીએ, તો વાઇલ્ડ લાઈફમાં ભાઈ કઈ રીતે વાઇલ્ડ લાઈફ ઉપયોગી થાય? તો તમે વાઇલ્ડમાં જે સંરક્ષિત છે એ તો સંરક્ષિત છે, પણ આપણી સાથે પણ એવી ઘણી બધી સ્પીસીસ છે જે આપણી સાથે જીવન જીવી શકે છે. હું એક લગ્નમાં ગયો પોરબંદર, લોર્ડ્સ નામની આવી હોટલ હતી, એમાં મારી ઉપર ગરોળી પડી એટલે ગરોળી મેં લઈ લીધી અને મેં જોયું કાં તો ગરોળી જુદી છે સાહેબ. એ ગરોળી જ્યારે આપણું એક્સપોર્ટ ઇમ્પોર્ટ વસ્તુ થતી હતી, આરબમાંથી એક ગરોળી આવી ગયા છે. પોરબંદર વિસ્તારની એક ગરોળી જુદી છે સાહેબ. એનું નામ છે ફણાવિલસ પોરબંદરેન્સિસ, એનું નામ અલગ આપ્યું. અને અગમ્ય કારણોસર પોરબંદરનો એરિયા છોડતી જ નથી, છે આરબ દેશની. એવા મુવમેન્ટો જો તમે જોતા રહ્યો. મારો એક મિત્ર હતો, એના ઘરે હું ગયો તો આગળના ભાગમાં ઉંદેડી મરેલી પડી હતી. એટલે મેં કીધું ભાઈ ઉંદેડી કેમ મારી નાખી? તો એ આખો ડિસ્ટર્બ થઈ ગયો કે સાહેબ તમે ફોરેસ્ટમાં આ કાંઈ ગુનો થાય? ખરેખર એને ઉંદેડી એના ઘરમાં આવીને મારી નાખી, ઉંદેડી બહાર ફેંકી દીધી. એ ઉંદેડીને મેં લઈ લીધી અને પ્રિઝર્વ કરી, સરસ મજાનો ફોટોગ્રાફ કરી. એ મેં ગુજરાતની અંદર નવો ઉંદેડો શોધ્યો, વ્હાઇટ ટેલ્ડ રેટ. એટલે મારો મિત્ર ડિસ્ટર્બ થઈ ગયો. તમારે જોવાની દ્રષ્ટિ કેવી છે? તમને એ ખબર છે કે તમે જે ઘરમાં રહ્યો છો ત્યાં 5 પ્રકારના કરોડિયા તમારે ભેગા રહ્યા છે? ના, તમને ખબર નથી. પછી તમે જે ઘરમાં રહ્યો છો ત્યાં તમે 3-4 પ્રકારની કીડીઓ રહે છે. હવે સાહેબ તમે એક વસ્તુ કહું કે આપણે નાના હતા, ખરેખર આપણે તો બાળપણ એવું ગયું કે છોકરાને કીડી કરડે એટલે છોકરું રાયડું નાખવા માંડે. હવે ક્યાં કીડી કરડે છે? કારણ કે કીડી તમારો રેઝિસ્ટન્સ પાવર ચેક કરતી હતી. હવે તમે કીડીનો જ નાશ કરી નાખો, માકડનો નાશ કરી નાખો, મચ્છર વાહે પડી ગયા છો, કોઈ જીવજંતુ તમારી બાજુમાં રહેવા જ નથી દેવાનું. આ વસ્તુ જે નાશ થઈ રહ્યો છે એ બચાવવા માટે થઈ અને આપણી આ બાયોટેકનોલોજી, આપણી જે ટેકનોલોજી, આપણી જે વિકાસ કરશું તો આપણે જીવી શકીશું. જેથી તમારી ભેગી મધમાખી અહીંથી જતી રહી, 15 દિવસમાં પૃથ્વી ઉપરની આખી પોપ્યુલેશન સાફ થઈ જાય. કારણ કે ઘઉંનું, મકાઈનું, બાજરાનું, તુવેરનું એ બધાનું પોલિનેશન મધમાખી કરી દે છે. હવે મધમાખી આટલું બધું ઇમ્પોર્ટન્ટ વસ્તુ છે. આવી તો આપણી આસપાસની અસંખ્ય વસ્તુઓ છે જે ખરેખર આપણે ગુમાવી રહ્યા છે. આપણે એક વીડીસાઇડનો સાહેબ ઉપયોગ કરી રહ્યા. કોઈને એ ખબર નહીં હોય કે 60% આપણી ઘાસ આપણે ખાસ કરીને કીડામારી નામની એક વનસ્પતિ થતી સૌરાષ્ટ્રમાં, રાજકોટની આસપાસ, આપણા જુનાગઢમાં. હવે ત્યારે કોમન રોઝ નામના બટરફ્લાય હતા, આપણે ગામના બધે બટરફ્લાય ફરતા હોય. હવે કીડામારીનો જ નાશ કરી નાખ્યો. એવી તો અસંખ્ય વસ્તુઓ છે. તમે સીધું જ દવાનો ઉપયોગ કરો છો, તમે સીધું જ ઝેર ફેંકી દો છો. તમે ગમે, હું અત્યારે આંબાનો નિષ્ણાત છું. અમે સાહેબ ટાંઝાનિયા સુધી ટેકનોલોજી શેર કરી. હું પોતે ફોરેસ્ટ ઓફિસર છું, પણ મને આમાં વધારે BSc એગ્રી નાતે. તો ટાંઝાનિયા અંદર સાહેબ આપણા ગુજરાતીઓને હજારો એકર જમીન છે. તમે કલ્પના તો કરો, અહીં જુનાગઢમાં નહીં એટલી તો ટાંઝાનિયામાં જમીન છે આપણી. અને એમાં એક મને ખેડૂત મળ્યો, નામ છે લાભુભાઈ ચાવડા, અને અસલ દૂધ વેચવાવાળા આપણા આયર બાપુ. તો મેં કીધું તમે મને કે સાહેબ રાજકોટમાં મજા આવતી નથી. સાહેબે કીધું 2 વાગે સુઈ જાય, 4 વાગે ઉઠવાનું, મારે સારું ભેગું થાય નહીં. એટલે મેં 10 વીઘા ટાંઝાનિયામાં જમી લીધા. સાહેબ અત્યારે આપ કલ્પના કરો, 35,000 એકરનો માલિક છે લાભુભાઈ ચાવડા. તમે જુઓ સાહસ કરવાની સ્થિતિ. તમે જેમ ટેકનોલોજી કરી, તમે જેમ સાહસ કરો, તમે ભણો નવું, ઉત્થિષ્ટ જાગ્રત પ્રાપ્ત વર્ણું નિબુદ્ધિ થા. ઉઠો જાગો, અને જેની પાસે જ્ઞાન છે એની પાસે જઈને બેસો, અને તમારો બેડો પાર થઈ જાય. માટે નચિકેતા બનો. નરેન્દ્ર મોદી સાહેબ કહે છે ધ્રુવ બનો, નચિકેતા બનો, પ્રહલાદ બનો. તમે સાહસ કરો, બળવો કરો, નહીં મારે શીખવું છે, તો તમે શીખી શકો. એ આપણા ધર્મગ્રંથોની અંદર આપેલું છે. એ જ ટેકનોલોજી બાયોટેકનોલોજી કે મેં તમને વૈદિકનું દર્શન આપ્યું. પણ સાહેબે જે આ, કારણ કે આપણે જે વિસ્તારનું અન્ન ખાધું, જ્યાં નોકરી કરી છે ત્યાં જો લાગણી કેવી છે. ગાંધીનગરમાં આવી મોટી આવી શિબિર થઈ શકે સાહેબ, હજારો સાયન્ટિસ્ટો આવે. પણ અહીંયા કેમ આવે? કારણ કે અહીંયા આપણે નોકરી કરી છે, આપણું અહીંયા ઋણ છે જુનાગઢનું. અને આ તો કેવું છે, સુદર્શન તળાવ અહીંયા 2500 વર્ષ પહેલાં બાંધ્યું, અશોકનો શિલાલેખ 3300 વર્ષ પહેલાં. આની અંદર કોક દીક તો જુઓ કે આમાં શું લખેલું છે સાહેબ. આમાં લખેલું છે 2.5 કિલોથી વધારે લાકડું તમે બાળી ન શકો. આ અશોકનો શિલાલેખ 3300 વર્ષ પહેલાંનો લખેલો છે આપણા વિસ્તારની અંદર. જ્ઞાન તો આપણે જ દીધું છે, બધું જ આપણી પાસે હતું. માટે હાલના યુગની અંદર આ ટેકનોલોજી આપણે ટ્રાન્સફર કરી, આપણી ખેતી આપણે ગુમાવી રહ્યા છીએ. હું એટલે માટે આવ્યો છું કે અત્યારે સાહેબ આંબા કાપી રહ્યા છે. હવે વિડીયા એવા ફરતા હોય છે કે ભાઈ આંબા હવે તો ઉત્પાદન, ભાઈ બાયોલોજીકલ ફેક્ટર તો કામ કરતું હોય. સાહેબે કીધું કે મગફળીનો ભૂકો, નીતિશ ધેરિયા સાહેબ કીધું ભૂકા વિનાના થઈ ગયા. તો ખેડૂતોને પાસે ખવડાવવા કાંઈ નથી. એ તો બાયોલોજીકલ ફેક્ટર છે, એને કેમ બચાવવું, એમાં ઢાંકવાનું આવે. તો એવું જ આ ઢાંકવાનું છે સાહેબ, કે ટેકનોલોજીકલી જીનેટિકલી ફેરફાર કરી આ ટકી શકે એવી સ્પીસીઝ આપણે લેવાની છે. આંબાની અંદર સાહેબ હું બહુ સારું નોલેજ ધરાવું છું, કારણ કે આંબામાં નાની એવી ભૂલથી માણસો, આપણા ખેડૂતો આંબો ગુમાવી દેતા છે. નરેન્દ્ર મોદી સાહેબ એવું કીધું છે કે 2 ગણી આવક. અમે જાડેજા સાહેબને મેં કીધું 2 ગણી નહીં સાહેબ, ગીર સોમનાથમાં 6 ગણી આવક ખેડૂત લઈ શકે છે અત્યારે આપણી ટેકનોલોજીને લીધે. કારણ કે તો શાંતિ ખૂબ છે, માણસ ગમે સ્ટડી કરી શકે. સરકારમાં તમે જરાક અરજ કરો ને સાહેબ તો સબસિડી મળે છે. આજે એક ખેડૂતને ત્યાં 20-20 હજારના સાધનો પડ્યા છે. તો પછી એને ટેકનોલોજીકલ ઉપયોગ કરી, આપણે થોડીક સ્ટડી કરી, આવી રીતે સિમ્પોઝિયમ કરી, આવી રીતે જીનેટિકલ સ્ટડી કરી, અને આપણી ખેતી જે મૃતપાય, કદાચ ક્યાંક નુકસાન થઈ રહ્યું હોય પર્યાવરણનું. હવે એમાં માછલા તો બધા સરકાર ઉપર, જેટલા સરકારને દોષિત કરો એટલો વિરોધ પણ સાચો ન હોય, અને જેટલી સરકાર એમ કે હવા કરતી એટલું બધું સાચું નથી હોતું. પણ છતાં સરકાર ખૂબ સારું કામ કરી રહ્યું છે. ખેડૂતોની સાહેબ ખૂબ, મેં જ્યારે આંબાની પહેલું પ્રોડક્ટ લીધું, જંગલ જેવું હતું લીધું, ત્યારે 200 રૂપિયા ભાવ હતા. આજે સાહેબ હું 4000 રૂપિયાના 20 કિલો આંબા વેચું છું. 4000 રૂપિયાના 20 કિલો. પણ એ તમે ઓર્ગેનિક કરો, તમે છાણ ઉમેરો, તમે ખેતીને શીખો, બીજા પાસે જઈને શીખો. મફત ટેકનોલોજી આપી. મારું એક ખેતર છે સાહેબ, એમાં હું મફત ટેકનોલોજી, હું પહેલાં એમાં સ્ટડી કરી લો, પછી ખેડૂતોને બોલાવું, જો આ મફત કરાય. હું પહેલાં બધું જ કરી લો, પછી ખેડૂતોને બોલાવું, જો આમ કાપો. અને આંબાને કાપી રિજુવેનેટ કરો. ભાઈ એક ડાળ તમારી તૂટી ગઈ છે, હાથ તમારો ખરાબ થઈ ગયો છે, તો કાપવો પડે તો નવો થઈ જશે આંબો. એ રીતે ટેકનોલોજી સપ્લાય કરી અને આપણે ખેડૂતોની મફતમાં ટેકનોલોજી, અમે 10 હેક્ટર 20 હેક્ટર સુધી મફત ટેકનોલોજી સપ્લાય કરે છે. ભાઈ તમે આવો, હું તમને શીખડાવું. તમારે આંબાની ત્યાં જાવ, તમે ખેતી કેવી રીતે કરવી, કેવી રીતે આંતરખેડ કરવી, એ પણ આપણે જ્ઞાન ધરાવતા નથી. કેરી ખાવાનું પણ આપણે જ્ઞાન ધરાવતા નથી. માટે આપણે જ બધું જાણી છીએ, એ વિસ્તાર આપણે છોડી દેવાની જરૂર છે. અને આપણે આવી ટેકનોલોજી ઉપયોગ કરી અને આપણી ખેતીને સમૃદ્ધ બનાવીએ. કારણ કે એટલે આંબામાં સાહેબ જે કીધું એ રીતે આ જીનેટિકલી જો સ્ટડી કરી અને આપણે આંબાઓને, આપણે નાળિયેરને, આપણે કેળાઓને આવી રીતે આપણે ઉત્પાદન કરી શકીએ. અમે સાહેબ અહીંયા હું જ્યારે ફોરેસ્ટ ઓફિસર હતો તે ઉપરના ભાગમાંથી સીતાફળ લઈ ગયેલા સાહેબ. એ સીતાફળ અમે 2000 સીતાફળ તાલડામાં વાવ્યા. સાહેબ તમે દુનિયામાં આટલા સ્વીટ સીતાફળ ક્યાંય નહીં જોયા. ત્યાર પછી મેં બીજો એક સ્ટડી કર્યો. હું જ્યારે અહીંયા આ વિસ્તારમાં ફોરેસ્ટ ઓફિસર ત્યારે હું 200 આંબા અહીંથી લઈ ગયો, ઉપર ટેકરા ઉપર, જ્યાં સાવ આંબા છે ત્યાં. એ આંબા વાવ્યા. આટલા તોકતાઈમાં પણ મારો એક આંબો પડ્યો નથી. પણ આવી રીતે તમે તમારી ટેકનોલોજી, તમે તમારો અનુભવ જો તમે સપ્લાય કરો તો તમે સારી ખેતી કરી શકો, સારું આર્થિક ઉત્પાદન કરી શકો. સરકાર, તમે પોતે તો સધર થાવ, સાથે સાથે સરકાર પણ સધર થતી હોય છે. આવલા વ્યાખ્યા જો જીડીપી એટલે શું, આપણે તો ઓહો જીડીપી કહો તો બધા ઊંચા પડી જાય. જીડીપી એટલે એક પ્રકારનું બંડલ ઓફ મનીનું એક્સચેન્જ છે. અહીંથી તમે 100 રૂપિયા દયા, 100 રૂપિયા સામે વાપરો એટલે જીડીપી ઊંચી જાય. સરસ દાખલો તમને કહું કે એક શેઠને ત્યાં નોકરાણી કામ કરે છે, 5000 રૂપિયા પગાર છે. જેવો શેઠ 5000 રૂપિયા પગાર આપે, જીડીપી અપ, ઇટ્સ અપ લાઈક એનીથિંગ. જેવો શેઠ એને 15000 રૂપિયા પગાર આપે, જીડીપી એનાથી ઊંચો થઈ જાય. પણ જ્યારે શેઠ અને કામવાળીનું જો લગ્ન થાય અને પગાર બંધ થઈ જાય, કામવાળી તરીકે ઘરવાળી તરીકે કામ કરે, જીડીપી ડાઉન. આ જીડીપીની વ્યાખ્યા છે. તમે એમાં કાંઈ મોટું કાંઈ છે ને. હવે આપણને કેટલું મોટું લાગે, ખરેખર જીડીપી બંડલ ઓફ એક્સચેન્જ ઓફ મની છે. એવી રીતે સાહેબ આપણી મને આ બોલવાનો મોકો મળ્યો. અમારા વડીલ, અમે વતનના છીએ, જાડેજા સાહેબ અહીં પણ નોકરી કરી ગયા, અમે વડોદરા પણ નોકરી કરેલી, અને સરકાર સાથે સમન્વય સાધી લોકોની કેવી રીતે સેવા કરવી એ ખરેખર જાડેજા સાહેબ પાસેથી હું પણ શીખો છું. અને સાહેબ એક ફોરેસ્ટ ઓફિસર તરીકે ખૂબ પાવર હોય, તમે રસ્તા બંધ કરી દેવાના પાવર હોય, પણ તમે પબ્લિકની સેવા કરો. સાહેબ હું એક જગ્યાએ નોકરી કરતો હતો ઉકાઈમાં. જ્યારે હું સવારમાં ઉઠ્યો ને સાહેબ, 75% બાળકોને પગમાં ચંપલ જ નહીં. એટલે મેં 9 મહિનાની નોકરી કરી, બધો જ પગાર એમાં હોયો છે, કારણ કે છોકરાના પગમાં ચંપલ જ નહીં છે. હું ફોરેસ્ટ ઓફિસર તરીકે બધું જ કરી શકું, એને રસ્તા અટકાવી શકું, રોડ અટકાવી શકું, લાકડા કાપડા અટકાવી શકું. એમાં મેં રિફોર્મેશન લઈ આવ્યો કે સવારમાં તમે જુઓ કે એક અધિકારી તરીકે તમે, મેં પછી ગણપતસિંહ સાહેબને કીધું, મેં કીધું તમે જુઓ તો ખરા આ બધી મોટી મોટી વાતો ને, અહીંયા આવો તો ખરા. આ તો જુદું પ્રભુભાઈ મને કે જો તમે સાહેબ બીજા 2 વર્ષ રહી જાઓ ને તો અહીંયા કોઈ છૂટાય નહીં. એટલો પબ્લિકમાં હું, કારણ કે મેં એક સાહેબ સ્કૂલની મુલાકાત લીધી. મેં કીધું ભાઈ પાણી કેમ પડે છે? મને કે સાહેબ નળિયા નથી. તમે કીધું ફોરેસ્ટ ખાતામાં આવું કેમ? જો તમે હૃદયથી તમે અધિકારી તરીકે કામ કરો સરકારની સાથે, તો કીધું નળિયા તો હું આપું. તો એમાં સાહેબ 2 પ્રજાપતિ આપણા ટીચરો આવ્યો. એ કે સાહેબ કડિયા તો અમે છીએ, તમે જો નળિયા આપો, 10 થેલી સિમેન્ટ આપો તો સ્કૂલ તો અમે બનાવી દઈએ. સાહેબ સ્કૂલ બનાવી દીધી. મેં પછી 100 થેલી સિમેન્ટ અને 200 નળિયા આપ્યા. અને તમે જો અધિકારી તરીકે સરકારની સાથે રહે, આપણે વિરોધ પક્ષ તો છે નહીં, કારણ કે અમારે તો સરકારની ઇમ્પ્લિમેન્ટેશન અને પબ્લિકનું કેવી રીતે સારું થાય, સરકારનું કેવી રીતે સારું દેખાય, સરકારને કેવી રીતે હેલ્પફુલ થઈ શકે, એ રીતે જો આપણે અધિકારી તરીકે નોકરી કરીએ, એટલે જ અમને સ્વીકારે છે. જાડેજા સાહેબને જ્યાં જઈને સ્વીકારે છે. આપણે પણ યાદ હશે, એસડીએમ તરીકે સાહેબે રસ્તો બનાવ્યો તો જંગલ ખાતું સૌથી પહેલાં આડું આવ્યું હતું. આ સાહેબે બીજો રસ્તો બનાવ્યો તોડફોડ કરી, કારણ કે એ જ કરી શકે. થોડુંક હાર્ડ પગલાં લેવા પડે. એવું સાહેબ વિજ્ઞાનમાં છે, એવું જ ટેકનોલોજીમાં છે. આપણે આ ટેકનોલોજી ઇમ્પ્લિમેન્ટેશન કરી તો આપણું જીવન સુધારી શકી, આપણું રાષ્ટ્રને સુધારી શકી. ભાઈ એક વડાપ્રધાન આજ છે, 25 વર્ષ પછી એ નથી, પણ આપણું રાષ્ટ્ર તો રહેવાનો છે. અટલ બિહારી વાજપેયી કીધું બધાય જશું, પણ અમારું રાષ્ટ્ર તો છે જ. રાષ્ટ્ર ક્યાં જવાનું? સત્યમેવ જયતે, સત્યમેવ જયતે, સત્યમેવ જયતે. જય હિન્દ, જય ભારત.
Thank you so much. And now proceeding it with Dr. S.K. Gupta, Scientist, F, Wildlife Institute of India, Dehradun. And he'll be taking up the topic, Advancing Wildlife Conservation Using Biotechnology. And once again, thanking our delegates who had made their presence at this very moment. Thank you.
So first of all, I'd like to thank GSBTM for arranging such a wonderful session here and inviting me in front of you. So I'm going to talk about some of the uses of the biotechnology in wildlife research and management, especially when we come across with the human-wildlife conflict situation, or when we get of any wildlife crime cases. So in those cases where biotechnology are molecular genetics is going to help the proceedings. So it is very, very crucial. So the picture which is given here, though it is our Lord Krishna idol, but it is the idol, it is the article which brought the first techniques of DNA extraction from the ivory. And before that, there was no any technique exist to extract DNA from article like elephant ivory. So which I did, before joining WI, I am basically right now posted in Wildlife Institute of India, Dehradun. But before joining Wildlife Institute of India, I worked in CCMB after completing my master in biotechnology in 2002. I worked with the Indian father of DNA fingerprinting, late Professor Laljit Singh. So I am the student of him and started my career with him. So first going to talk about the use of molecular genetics, use of biotechnology, especially biotechnology is not restricted to genetics. There are various fields of biotechnology, but here we are basically focusing on the molecular genetics. So what is very, very crucial here, when we come across, when we collect the sample from the field, basically we cannot capture the animal, wild animal. So we rely upon the remote samples, we rely upon the non-invasive sample. And when we get the non-invasive sample, especially non-invasive abundant sample is the fecal sample, they are dropping. So when we collect the dropping, so that we can answer the various questions, especially first we accurately identify the species, which species this belong to. And once we collect the multiple sample of the one species, then we come across, when we start, then we start our investigation. We do the population genetics, we do the corridor linkage analysis and all those using those remote samples. So there are various way of doing that, and I am going to just briefly discuss about few of the findings here. Hog deer, which is Axis porcinus, very, very peculiar, very, very famous deer, and people know who goes into this area. It is found in Tarai Arc landscape. So hog deer occurs in west in the Punjab to northeast up to Manipur. So earlier it was thought that all the hog deer found in India were same, because they look same, they were categorized as hog deer. But when we compared their genetic sample, especially their dropping, their fecal pellet, and with the fecal pellet we came across that the hog deer occurs from the Punjab to Kaziranga, Assam, are the one Axis porcinus one species, one subspecies, but the hog deer which occurs very nearby to Kaziranga in Manipur, in Kebulamjo National Park, that hog deer is totally different. And that is the rare subspecies of hog deer which occurs from the Manipur to eastward. And now in the Southeast Asian country, hog deer population is almost gone. So we discovered with this technique that hog deer which occurs in Manipur is a very pristine and very crucial species, subspecies for conservation. And Manipur is also known for that. Kebulamja National Park is also known for one of the only population of the Elf deer, dancing deer, Sangai deer. So with along with the Sangai deer, the hog deer occurs in Manipur now become very crucial and very important for the conservation. So that we only came to know through using this technique, and we published in a scientific report. We also discovered, earlier we thought that there are various species, in fact, species of the musk deer, Kasturi mrig. But the musk deer, earlier we thought that which occurs only in Kashmir, they are called Kashmiri musk deer. But through our study, we came to know that musk deer which found in Uttarakhand as well as in Nepal, they are also the same species which is found in Kashmir. Earlier, those musk deer were called as Himalayan musk deer, but we never found Himalayan musk deer in this area. So it is the range expansion of the Kashmiri musk deer, or we can say that Kashmiri musk deer were wrongly named as Kashmiri. It is basically the standard population of the Himalayan musk deer. So it is again a scientific debate which our study using the molecular genetics opened up. And all these research which I am talking about, it has led to the getting the PhD degree of my students. So basically the first authors and first the researcher of my PhD student. This is very common bird species in Himalayan area, blue-throated flycatcher. So blue from the east to west. But when we collected the multiple sample of the blue-throated flycatcher, we came to know that blue-throated flycatcher of the west and even in the east. And in the east, in the same hill, like if you consider this Girnar in one hill, so the basement of the Girnar and top of the Girnar, the blue-throated flycatcher which occurs in the top and bottom were totally different. And that only came to know with the using molecular technique, using the population genetics technique. And the blue-throated flycatcher which occurs at the top and which occurs at the bottom were separated about 2 million years ago. So they have separated a different subspecies of the blue-throated. Earlier with our even renowned ornithologist, Dr. Trevor Price, who is working at University of Chicago, he used to come India on regular every year. So he thought that all blue-throated flycatcher is same, but our study, he is also one of the collaborator in my work. So he then got surprised, oh, we have such a very minute information about the single species at the different scale. So that only we can get to know when we apply the molecular genetics technique. Indian star tortoise is one of the heavily poached live animal. So world largest live animal, illegal live animal trade is about the Indian star tortoise. And one of my researcher, Dr. Subhasri Sahu, she completed her PhD. She collected the biological sample from the Girnar, from the Tamil Nadu, from the Andhra Pradesh, from Punjab, and we collected all sample from all the area, multiple samples, and we created area specific or at least their genetic profiling, so that if live animal is captured or caught at some of the point, so we especially came to know that which population it belonged to. And if we know this information, then only we can precisely rescue and release that animal in their habitat. Otherwise, what will happen, if any consignment about 1,000, 2,000 tortoise is captured in Ahmedabad or in Mumbai, if you bring all the animal and just release in Girnar, so it will hybridize, it will contaminate the population, native population of this area. So if you know the accurate information through their genetic study, so we can specifically release the animal in their proper habitat. So this baseline information is now available with WII, and this information is now used in identifying. We have done one consignment from the Mumbai custom. So they caught the animal. We identified that some of the animals were from the Gujarat and some of the from the other areas. So their proper release decision can be taken after that. So horseshoe crab, which is again very important species, which is used in clinical research also. Their blood is used. So horseshoe crab genetics also we have done in our lab. One of the student completed his PhD, so we collected the sample from the Bay of Bengal, and we found that yes, in Bangladesh as well as Indian population are the similar then, but they were totally different from the population found in Malaysia, Thailand and other area. So these are the way how can we identify and how can we use advancing the wildlife research. But it has also used in taking up the challenge nowadays. Very big challenge is the human-wildlife conflict. And in human-wildlife conflict, when such a conflicting animal is reported, so first of the objective is to identify the species from the remnant which is available from the carcass, from the human decayed body. So there you can easily get the hair sample. And with the hair sample, first we identify which species was involved in that conflict using DNA test. And morphology also there, but DNA is more precise. And if we collect the sample from multiple locations, multiple incidents, then we can get to know that in all the incidents, though they are the same species, whether both are multiple samples, multiple incidents is done by single individual or not. So we can do with the DNA fingerprinting, we can identify the species. And in future, if animal is captured or eliminated in that area, so we can match again the sample of that eliminated animal and compare with the previous instances and say that yes, we have accurately caught that culprit animal. So it is very useful in identifying multiple cases we are doing on routine basis in our lab. And not only for the human as a victim, but sometimes animal is also victim. In one day, we have received information that in a night, 3 leopards found dead in Rajaji Tiger Reserve. So their gut content were collected and sent to us, and all the leopard gut content were filled with the dog. And then we compare with DNA fingerprinting, all the gut content dog was the single individual. And in the same area, dog carcass, half-eaten dog carcass was collected and sent for the toxicological study, and found that that dog carcass was fully loaded with the heavy metal pesticide. So dog was poisoned to kill the retaliate killing for the leopard in that area. So such things are also happening, and all these things which I am discussing here is the part of the some of the scientific study in paper. So we have done though more than 5,000 wildlife offense cases, 5,500 cases we have done so far from all across the country, and many cases we have received from the Gujarat also. Gujarat Forest Department is also continuously sending their sample to our laboratory in Dehradun. Every year we do receive 250 to 300 cases in our laboratory, so it accumulates the number. So that we do using 2 different technology, morphology as well as DNA, but DNA is very prominent. About 65 to 70% cases are required only DNA test. So here preservation was a problem, so alcohol is the best preservative, which I explained to everywhere, not formalin. Never use the formalin as a preservative for the DNA testing. Silica is also one way to collect, but we never get the sample in the blood sample in the vial, in the wildlife crime cases, because in the vial we can only get the blood from the live animal, and in wildlife crime cases animal is dead. So what we get, the blood stain, blood clot, blood stain on the weapon, leaf, log and litters. So those blood stain we use to identify the species using the mitochondrial DNA sequencing. We have done multiple cases, many cases. I have signed myself more than 2,500 wildlife forensic report. Many of them are very, very essential and important, and many of them got published also in as a case report. But one of the very interesting case I am discussing here, the murder, not hunting I would say, murder of 2-year female tigress in Hyderabad Zoo. It happens in year 2000. And next 5 years, all agencies and all investigating agencies were behind it, but no clue they got that where the sample is gone. So their skin and claw were taken away. But after 5 years, one local boy was caught, and with that sample, he was wearing a claw pendant in silver frame. And that claw, I extracted DNA, first of all, extracting DNA after 5 years from a sample which is routinely used as a pendant, so very, very challenging, but we could extract DNA. And with the DNA test, the question was asked whether it is the same tigress which was killed in the zoo. So though luckily this tigress was captive born, so we collected their relatives' blood, mother, father, and she was born with the brother sibling. So all the 3 individuals' samples were collected, and then I made the DNA fingerprinting profile using mitochondrial DNA as well as the nuclear DNA, the actual microsatellite loci. And based on final study, we could conclude that yes, it is the claw were from the same family from where one tigress was missing. So we concluded it. There are many cases. In one case, a leopard was killed by burning alive. So in that situation, ashes were collected. Ash samples were collected, and those ashes were sent to me, and from the ash we could identify that yes, there was a remnant of male leopard. So we concluded that report. We do lot of training and sensitization program. We have been invited in Interpol headquarters to teach, Interpol headquarters in France, to teach the European participants how to differentiate shahtush shawl from the non-shahtush. Shahtush is a very fine fiber coming from the Tibetan antelope, chiru. So it is contraband items, and Interpol is behind it. They are also looking very seriously in control of illegal wildlife trade, because somewhere the linkages of illegal wildlife trade is with the terror funding. So they want to look at that terror funding angle. They want to stop that. So that is why they are aggressively looking to stop the illegal wildlife trade. So we went there and explained the European participants how to differentiate shahtush from the non-shahtush pashmina shawl. Tiger stripe is very, very unique, and it has been used in identifying the individuals, especially if you see here, this tiger was photo captured in Pest Tiger Reserve, and its skin was 3 years later found in Kathmandu. So because of their stripe pattern, we could make it out, yes, it is the same tiger which was captured 3 years back in the page. So we can pinpoint, identify that which are the tiger reserve is in poaching pressure. So we can enhance the enforcement, we can enhance the protection there to protect the species. Dead animal is no good for the conservation. We need to protect them before they get killed. So that is why the tiger number has increased from 1411 of 2008 census to now the recent census of 2022, the tiger number is more than 3,600. So though our Honorable Prime Minister set a target to increase the number in by 2030, but we have achieved that target more than double much before the given target time. So in 2022, it was more than the double number. And we provide the training to various agencies, scientists of the Forensic Science Laboratory, scientists from the Forensic Science Laboratory of the neighboring countries to get to know how to wildlife forensics is done. The wildlife forensic science and this and forensic science, both are totally different. In forensic science, we only focusing on the one species, that is human. But here in wildlife forensic science, there are thousands of species together. So we are developing the science with the R&D, as well as we are also providing the enforcement support, scientific support to the agency working all across the country. So that is the outcome of our lab. We have the good laboratory, good facility and everything. Constraint is always there. And that's what I would like to tell you briefly in this presentation. Thank you all. And open for discussion. If you have any questions, you are welcome. I completed my PhD from Gujarat, Saurashtra University. So Gujarat is connected to my academic journey. Yeah. Any doubt or any question in audience? Yeah, so ancient DNA is again another topic where many scientists are working, especially when I was in CCMB, one of my junior, Neeraj Rai, he is in Birbal Sahni Institute of Paleontology in Lucknow. He is working on the fossil DNA only, so he is my colleague, my junior and collaborator also for some time. So yes, we are working. Archival DNA is very, very essential, very, very good, especially the sample stored in the museum and in the collections, the princely collections. So those are very good sources. Like though cheetah were extinct in 1960, 1950, but their samples are still, old samples are still available as a form of the taxidermy. So those can be used for comparing the recent cheetah introduced here in India.
In wildlife conservation studies in BKNM University. मेरा एक छोटा सा बेसिक क्वेश्चन है कि सपोज एक कॉन्फ्लिक्ट सिचुएशन है कि लोकल्स ने हमें बताया कि मेरे पे एक कॉन्फ्लिक्ट अटैक हुआ है, लाइक लेपर्ड ने अटैक किया है मेरे पे, और हम लोगों को लगता है कि वो अटैक उसने सिर्फ कंपनसेशन लेने के लिए किया है, राइट? सो व्हाट आर द बेसिक रिक्वायरमेंट, और ऐसी कौन सी चीज़ हम लोग वहाँ से कलेक्ट कर सकते हैं फॉर दी एडवांस एनालिसिस और दी लाइक डीएनए सैंपल और एनीथिंग, और कितने टाइम तक वो वैलिड कंसिडर की जा सकती है और कैसे सैंपल ले सकते हैं?
इट इज़ वेरी वैलिड क्वेश्चन, वेरी गुड क्वेश्चन. In fact, basically in human wildlife conflict, wildlife will not turn up to explain them that they are innocent. So we need to take the advantage of the technology available here. So basically, lot of murders have been given in the court of the that it was killed by the animal, and forest department under pressure, they have to provide a timely compensation. For them also it is a challenge. some, and maybe sometime or frequently they also get compensation for those cases which was basically a law and order problem. So especially in those situations, if people are claiming, those who are claiming he is already dead, I think, he is no more. So there, from the decedent, you can collect the evidence, you can collect the sample, like if anybody claiming that that person is molested or killed by the wild animal, so you will definitely get some of the evidence of the wildlife, especially on his body, if it is a tiger or leopard or lion, so their hair get very easily studied. So their hair definitely will be there on the carcass, on the body. So you need to collect those hair and get sent to laboratory for the confirmation of the species. As well as if there are bite mark, so there are very peculiar pattern with the attack by the cat and attack by the other animal. So cat, when it attack with a lion, tiger, leopard, all cat has very habitual form of the attacking. They capture from the neck and try to break the trachea and puncture the jugular vein. So you will see in that situation the puncture mark on the neck, and that if somebody can make making hole using the weapon, but you can make it out whether it's artificial punctured, not you, at least the doctor who is doing the postmortem can do the further investigation and give the information report whether that puncture mark is by the any weapon or by the some other natural way. And when animal consume the body, so they open the carcass like anything. However, if it is a murder situation, so the cutting will be very sharp. So those are the circumstantial evidence from the crime scene. Only with the help of the forensic expert and team can get to know that what was the situation actually, and they can assess and give the report. Any other? Yeah. Yes, yes, yes. If you get the lick mark on the body, licking mark, so you try to get the swab from the licking mark, not from the wounded side, because wound if there are, so there will be more the DNA of the human. Yes. So you try to avoid the area where it is opened and there are wound and blooding. So you collect the area, you try to find out the area where animal licking mark is there, saliva mark is there. So you take the cotton swab with the wet conditions and collect the sample and send to
because
laboratory.
in the field area, he is also saying, so many times in this periphery of the gear, so many times happen that type of cases. So many times in man-animal conflict also, the enough sample of the hair sample is not available. Only source is for the DNA is the salivation. If possible, because of the in the cases, other circumstantial evidence is also not available. So that cases is saliva, if possible, from the saliva elevation of the DNA.
Yes, yes, it is possible. It is possible.
And even in the compensation matters, so many times, so many complications are arise. Owner is saying that's killed by the wild carnivora, but there may be sometimes dog also kill the small calf or buffalo calf, cow calf. In that cases also, this type of cases has arises. If from saliva is possible, then it's beneficial.
It is possible. And if dhole and wolf are attacking, so their eating pattern is different than the cat. So it can be even identified. A good ecologist can judge the carcass and tell which animal, which group of animal at least attacked here. It is possible.
Hello, sir. Sir, in one of the slides you have shown that stripes are the unique distinguishing marks. So on the face of the tiger or the tigress, one arrowhead or the W-shaped mark is there. So was it considered to distinguish in that?
Any mark, any stripe mark, whether it's on tiger, whether it's on leopard, their rosette pattern, they are very unique. Even on the face, stripe is on the face also, so that also considered as a...
Sir, in that particular case, when the tigress was found from the Kathmandu, was it considered that it was the same tigress or the different?
Yeah, it was considered as same tigress which was captured in Pench. because it has full, when we place the camera trap, we collect the sample from both the side at a time. We put the camera on both the side. So if you get the sample from the left, and next time tiger is moving from the right direction, so you will consider two different tiger. But if you collect the sample simultaneously from both the sides, so you will get the comprehensive picture of the one tiger stripe. So that's how we set up the camera trap in the ecological study. So we get all the information.
Thank you, sir.
Any other question? So I think no question. So we can wind up. Thank you.
Thank you so much. And now moving ahead with the next speaker, that is Dr. Apurva C. Puwar, Scientist B, Gujarat Biotechnology Research Center, Gandhinagar. And he'll be conducting the session on GBRC activities in wildlife genomics.
Very good noon, dignitaries, friends, students. So I'm Apurva, working as Scientist B with GBRC. So at GBRC, we are working on various aspects, including wildlife. So wildlife is one of the pillar considered at GBRC. So we are working in virology. We are working on One Health concepts. We are working with ICMR, DBT, national level institutions. As a part of wildlife or the projects which are being undertaken at GBRC, like includes seroprevalence study of pathogens in Asiatic lion. So we work closely with Forest Department for the prevalence of CDV, Babesia, as well as Anaplasma, vaccine development against life-threatening pathogens, which considers humans as well as animal vaccines. Barcoding of flora and fauna of Gujarat, that work was undertaken at GSBTM, which is now converted to GBRC. Adulteration of herbal formulations, that work also we are using as a molecular tools and technique. And the one of the beautiful project that is being funded by NOHM, that is National One Health Mission, and ICMR, that is detection of zoonotic spillover in increased bird-human interaction or the interface settings using One Health approach, where we are covering bird sanctuaries and the wildlife settings. So to start with, during the GSBTM time, which was like the research activities was established as a Gujarat Biotech Research Center in 2018. So before that, we completed with the collaboration of Sakarbagh Zoo and the Forest Department the first mitogenome of Asiatic lion. So the discovery led to 17 kb mitogenome of Asiatic lion with annotation and all, and found that the divergence of the Asiatic lion from the leopard species was happened before 2.7 million years ago. Then we were included in the seroprevalence studies in Asiatic lions. We all know that the efforts of the state government and the forest department has led to increase in the population of lions, the pride of Gujarat, from 674 to 891, and even from year 2000 to year 2020. Increase in the lion population. So GBRC contributed in the outbreak of 2018, and during that time GBRC received more than 400 blood serum samples from the entire century, and each and every animal was tested for antigen antibody test. As well as we developed a very rapid method for identification of CDV, Anaplasma and Babesia, and the tests were carried out during that during the 2018 outbreak. So GBRC successfully sequenced the CDV genome and found that the 2018 strand, which was the causative agent for deaths of lion at that particular time, was having fusion protein with the highest number of mutations, which is the causative agent, and the neutralization test against the vaccine was not proven. So GBRC has started sequencing the more number of samples, and then based on that, the inactivated vaccine development was taken care. So which is currently now under mice model testing, which would be taken up further for the feline and the canine model testing with the permissions of the higher ups. So the vaccine developed is a common concept of inactivated vaccine, where we are using different kind of adjuvants and preservatives. So second approach also we have taken, that is adenovirus based mRNA vaccine. So the same platform GBRC has developed. So the group at GBRC is working on the development of adenovirus based vaccines, where CDV is also considered, influenza, likely highly pathogenic influenza strain like H5N1, H3N2, as well as SARS-CoV-2, or the concept is to develop the vector or the technology which you can readily made any kind of vaccines, mRNA based vaccines. So that technology GBRC has also worked. Then in the previous year, like since 2007 to 2014, GBRC as a GSBTM has worked on barcoding on various species. So it is very happy to contribute for the barcoding at GBRC, like bacteria 16S we have done, fungus ITS, animals UI, plants we do have done RBCL, METK, TRNH, PSB and YCF. This marker already done, and you can see that almost entire Gujarat has been covered, covering almost unique 3,000 species of all three kingdoms, animal, plant and microbes. Then the one concept was DNA based authentication of herbal formulations. So as per the Ayush department's interest, so the work was carried out to identify the adulteration made in the tulsi, different kind of tulsi, different kind of ashwagandha, or like people are mixing this papaya seeds in carica kind of thing. So GBRC developed and patented this technology to identify whether what kind of adulteration are being done, and you can taste even in the formulation and even in the raw material taken from the forest or taken from the any farming settings, like whether whatever raw material is there is adulterated or not. Then this one is ICMR Bird Sanctuary project, which is mainly for the detection of spillover agents with respect to human environment interface. So here we are targeting so many samples, like where what we are doing, we are emerging zoonosis pathogens from almost all the three bird sanctuaries from Tamil Nadu, Sikkim and Maharashtra. So you can see that each and every samples collecting from all three states are finally reaching to GBRC for sequencing and for analyzing any viral pathogen or detecting any spillover events happening in three of the states. So like this one is the representative graph that shows like GBRC is one of the state agency which is in collaboration with more than 10 institutions at national level is working, and we are following the workflows for NGS. So recent study that we completed was from the Sikkim, that rhinovirus, which is very well known for common cold or very common virus, that also was found from certain bird species and all. So these are the representative graph for viral detection from the NGS data. Then Gates Foundation funded surveillance of influenza, LSDV, FMDV, work that Dr. Kanani also presented. So GBRC is also collecting samples from almost 7-8 sites and almost found more than 100 samples positive for influenza. And we are also looking for the HPI as well as LPAI strains, like what kind of highly pathogenic influenza are present in these particular sites or not. Soil, water and air can give you a very much advanced signal what kind of viruses or what kind of pathogens are present in the forest or nearby forest, and what kind of human pathogens are entering the forest, and what are the boundaries are being crossed by particular microbes. You can very well detect them. So this one is for the students sitting over here, like GBRC's shared laboratory facility. So state government's initiative to provide state-of-art facility laboratory, like whatever next generation sequencing instruments, RT-PCR, qRT-PCR we do have. Even the computational facility are available at GBRC at very subsidized rate, like it almost 50% subsidy is given all to students, and for all university and staff members, like the faculty members, it is cost to cost. So these are the like more than 40 instruments are there, and few more are being added to GBRC's catalog, and till now more than 17,000 usage of this particular instrument is being done by the external users, totaling more than 1,400 people and 285 organizations. Thank you. Questions?
સર આ ગીરમાં વારંવાર જે ફોરેન્સિક રિલેટેડ જે કેસીસ આવે છે એમાં મેઇન જે પ્રશ્ન આવે છે કે કોઈ પણ પાર્ટ્સ મળે બોડીના ત્યારે સ્પીસીસ આઈડેન્ટિફિકેશનનો હોય. હવે જ્યારે ફોરેન્સિક રિલેટેડ હોય છે તો એક એ પૂછવું છે કે GBRC રિપોર્ટ છે એ જ્યારે કોર્ટ મેટર થાય છે ત્યારે એ વેલિડ ગણાય છે?
GBRC is not generally entering into any court matter related identification basically. But GBRC is having strength to analyze like what kind of forensic cases are there. So GBRC is not particularly doing that kind of work.
Because in that type of cases, what happen? Sample we are sending in the CCMB or the WI Dehradun. Whenever sample result came, at that time simultaneously other related evidences are there. There may be manipulated or any losses are there. So many times, presently, so many times happen. Fake clothes are more frequently we are collecting. Fake clothes are even in the net marketing also, so many times selling is there fake clothes of the tiger and lions are. When they are captured and that type of issue arising, in that process so many long time cases pending for that result, and that times also so many time department have also lost, and so many times their culprit is go to the way of the escape from this case. That's why I'm
so same in the same line GBRC has worked in past, like many of the scat samples of man-eater leopard was handed over to GBRC and the same was reported back to forest department. So that work we have done from the scat specimens, and whatever evidences we get from the molecular technique that were handed over to this forest department. Similarly, vultures and some of the peacock killing cases were there with GBRC that were also responded back to forest department time to time.
Okay. But in the court case, there is if a report is sent, then and then legal process can go through. So that's why.
Court case.
department.
Okay, so thank you.
Thank you. Any other question? Thank you.
So altogether, it was a very knowledgeable session. And before we officially put a rollover of the vote of thanks, I would like to really make a mark asking that how many of us have taken down the notes that all our technical officers have put it in the frame for all of us. Can we get some hands raised upon? Then here's a huge round of applause to all of you as you have given your 200% for this session. And now let me call upon stage the manager GSBTM, Dr. Mitali Dabi, as she'll be taking the official vote of thanks for all of us.
Thank you, Ridhima. So, honorable dignitaries, guest experts, and all participants over here, very good afternoon. On behalf of this Gujarat State Biotechnology Mission, it is my privilege to offer vote of thanks for this very engaging and informative program. First and foremost, I would like to express our sincere gratitude to our Honorable Vice Chancellor, Sir, Honorable MLA of Junagadh, City President of Junagadh, Dr. Nala, for their invaluable presence and informative address. I would like to thank Mission Director GSBTM Jadeja Sir for his guidance and motivation to do this program here in Junagadh. We are incredibly thankful to all distinguished speakers, Dr. Amit Kanani, Dr. Gupta, Dr. Nishit, Jadeja Sir, Dr. Apurva for sharing their expertise and insights. Special thanks goes to Jani Sir to propagate this information and send their students. Sincere appreciation goes to GSBTM team for putting together this planned program. Thanks to the press media for full coverage of our program and to propagate this information among the state. I would like to thank our dear participants for your presence and active participation. Our commitment to wildlife is what makes this program more beautiful. Thank you once again and see you tomorrow in one more important topic of horticulture. Thank you.
So firstly, एक announcement है. Announcement A है कि if by any means or chance we are left out in taking down the notes, the entire session is on YouTube. We went live on YouTube. So all the participants can mark down that session from the YouTube as well. And secondly, before I mark a closure for and proceed towards the lunch, thank you once again. It was indeed a very informative session for all of us. Indeed, participation of yours made this wonderful day so very special. And nevertheless, all the gratitude and thankfulness to all our delegates and dignitaries, those who were present here, made their gracious presence. By some commitment, they had to leave early, but then again, a gratitude to all of you, those who have made their time and made this wonderful day so very special for all of us. Yet as Dr. Mitali Dabi requested that tomorrow we have yet another very knowledgeable and impactful session on horticulture. I want those who are definitely interested in that should mark their presence tomorrow as well and definitely mark down the notes for tomorrow as well. Thank you once again. This is Ridhima Majumdar signing off for the day. It was indeed pleasure seeing all of you. Thanks a lot, and we all together can proceed for the lunch. Thanks a lot once again. All the students, all the participants are requested to move towards my right hand side direction door. आप सभी का lunch वहाँ पे accommodate किया गया है. Towards my right hand side direction.